Okay, good morning, everybody. Welcome to day 3. Is it day 3? Yeah. I hope you all enjoyed the dinner last night and the after dinner and the after dinner, which seemed to continue. In fact, I think some people are still there. So we have a great morning for you. So the highlights today in the general session, we've got the Young Investigator Projects, the Shark Tank, which is now really popular, and there's an interactive voting sheet in front of you all with a QR code for voting in that session. And then there's a sort of final session on the importance of caution in the interpretation and conclusion of some clinical trials. So 9:25 is the Shark Tank presentations with 4 eager, talented young colleagues. And as I say, the voting system is a new feature. I think I just saw the, was it just, yeah, look at that, Rick. Wow, that's amazing. Wow, that's really good. I think that's the best photo ever. We need to send that to Birch, don't we? Yeah. It's a great place, yeah. Then the Shark Tank winner will be announced today at 12:15. So that's the plan for the morning. Rick.
And I'm up next. Continuing ed. Just see a show of hands. For who in the room will be applying for continuing education credits? Okay, it's good. So probably over half. We've gone back and forth over the years. Should we, shouldn't we? Is it worth the effort? I guess it's worth the effort. Yeah. Is that some of the companies? Some of them. Although Liz has been able to get money out of anybody, Tussing, so. Thank you, Pharma. Yeah, we'll talk about you in just a minute. So you got an email about with the disclosures and the link for the evaluation to complete after the meeting concludes. And now we also, in addition to CME for United States physicians and healthcare professionals, we offered for international CEs as well, and other and certificates for other attendees. That's a good thing. All right, next.
Okay, yeah. So just this list of colleagues. It takes a lot of people to make the meeting so successful. Their names are all listed here. So I think one final loud round of applause for all of the colleagues. So Gil, yeah. And obviously Liz, Donna, Grace, and all the colleagues listed here. Just give them another round of applause.
Okay, so now we're ready to talk about Liz. She's been threatening to retire for a few years. She said, "Dr. Barron, it's coming. I'm really going to do this eventually." But I think it's actually really now happening. So Liz joined the NMSG in 2018, and a lot of the metrics that happened in the NMSG under her administrative leadership are on this slide. It's pretty phenomenal, the numbers, and she is what I've learned about. What I've observed about Liz is that she's able to do all these things and get to all these incredible metrics seemingly effortlessly. And I know it does take a lot of effort. I know it does, Liz, but I never know. And it's just one of the hallmarks of a great partner in pulling something off like this, and how effortlessly she seems to pull it off. It's just incredible. So she is turning administrative leadership over to Donna. We'll hear about Donna in just a minute. She has told me she's going to stay involved some. What some means, we haven't defined exactly yet, but so we're really happy about that. But she's got things to do this year. Her daughter, Sophie, who's in the back there with her fiance Scott, wave Sophie and Scott, and husband Brian. I guess Sophie and Brian are getting married in a few months. So,
She's not marrying her father.
oh, I said,
Yeah, it's not that kind of marriage.
yeah, Sophie. I had it back, yes. You know what I'm talking about. So there's a Polk wedding in a few months, and we're very happy about that. So let's all thank Liz now and multiple times throughout the day and in the future for everything she's done to make the NMSG what it is today. Thank you, Liz.
So then we're really delighted to welcome Donna, who I think most of you will have met. Just a bit of background about Donna: Bachelor of Journalism from the University of Missouri, Master of Arts in Public Relations from Webster, extensive domestic and international meeting planning experience, which is really evident to us how quickly she's hit the ground running and working really well. Worked in sports public relations for the majority of her career, including owning her own company, where she managed the careers of 4 NBA basketball players, but still managing Rick is tricky. And she's a novelist, so now working on her 11th book, which will be published at the end of the year. So a very warm welcome to Donna, and thank you.
Okay, so once again, thanks to our chairs of this year's planning committee, Valeria and Emma. The amount of work that both of you and the committee puts in to get this to happen every year is extraordinary. And Mike and I really, really appreciate it because we do very little to get this to happen. Yes. Valeria, we were just thanking you, Valeria, again, for chairing the planning committee. And next year, Vino is one of the co-chairs. Is there another one? It's not 2 of them, just one? So Vino in London is the chair of the planning committee, and the committee has pretty much been established, although we had one person volunteer that wanted to be on it this time, and I think we're going to make room. So we love volunteers. So thank you again for this year and for if you're going to be serving over the next year. Thank you very much on the planning committee.
So then, once again, just a huge thank you to all of our great sponsors for this year. Just a reminder that it was sold out in terms of sponsor spaces this year. So if you're a sponsor, be sure to book early for 2026, and you're very welcome, obviously, to join us next year. But thank you to all the sponsors. So just moving through the sponsors once again. A big thank you to all of them: Platinum level, Avidity, Johnson & Johnson.
And then at the gold level, once again, Alexion, Arginex, Catalyst, Merck, Sanofi, Sarepta, the coolest name, Scholar Rock, Cartesian, Immunovant, NHTI. Next slide.
And then the bronze level sponsors: Dyne, NMD Pharma, Solid Biosciences, ML Bio, Amgen, Giverna, Octpharma, Lupin, GBS, UCB, Arthex, and PepGen Bio. So thank you to all of the sponsors.
I really like how we now have the opportunity for our sponsors to sponsor one of the events at the meeting. And when I just walked in for breakfast, wasn't Merck's name on the door? So anyway, we'll keep figuring out opportunities for the sponsors so you can get your name highlighted at different featured events during the meeting. I think that was another great Liz idea. So, okay, next year, I'm really excited about this. You know, I don't think we have a standard operating procedure for a lot of what we do, including picking the next site for the meeting, and it's an iterative process. A couple years ago, we got to Terrytown last year because I had just randomly remembered that Hiroshi Matsumoto did an ALS meeting at Terrytown like a decade ago, and it just occurred to me, and we looked into it, and it turned out to be a great place. I was in San Antonio, where I lived for many years, and for a meeting a couple years ago, and I stayed down in downtown San Antonio at the Menger Hotel, and I kept thinking, I bet this place could accommodate our group, and I think people who have not been to San Antonio would really like that, especially our London friends. You've never been to San Antonio. So we are going to San Antonio in September 2027, and I'll go to the next slide. So this is a picture of the beautiful Menger Hotel, founded in 1859. 26? Oh, yeah, 26. Yeah, I'm making a lot of mistakes. Husbands, future son-in-laws, years. Okay. All right, so the Menger is a very cool hotel. How many of you have seen one of the greatest TV shows ever, Lonesome Dove, the Larry McMurtry picture? Tony is. So there's these great scenes in the first couple episodes when Gus and Cal are getting to do their cattle drive up north, and they're in the Menger Hotel, in the bar in the Menger Hotel. That bar is still there. And it's also where Teddy Roosevelt launched his Rough Riders campaign. And it's just an amazing historic hotel. And it is virtually next door to the Alamo. So the Beaver is going to Texas next year. We've tried to get the Beaver to Houston a couple times. Aziz tried, it never really worked, but now he's going to go to San Antonio, and that's going to be exciting. So a couple other things that I just want to remind you of. Yesterday we talked about structure of the NMSG and how to be a member, you're a site membership. So I just want to remind all the leaders in the room at your sites that when people come and go, which happens every year in our units, let Donna know so she can update who is on your team as an NMSG. So not only so they know that they're a member, but we know and we can get emails out to the right people, which is important. So that's one thing. The other thing I wanted to say before we launch into today's event is the Rick's Real Neuromuscular Friends Neuromuscular Journal is still going somehow, the abstracts, as you know, are published in there. We once again want to encourage any of the junior investigators who are willing to send send a paper in from their abstract into the journal. That would really be welcome. So if you're interested in that, contact me. And then, am I still doing this, or do you want to do this?
Okay, so I think...
Yeah.
So, next year's venue looks really, really great, actually. So now we're very pleased to welcome our first presenter of the day. Sorry, the bosses need to speak.
As we said, no standard operating
Yeah.
procedures, right, just fluid. Good. And so will you introduce the first?
You can introduce. So, okay. So as co-chair for this year, we want to really bring Liz back. Come up here. We have a little token of appreciation for you from all of us. And I have to say, we've already heard about all your magic that you do, but personally, also, you make it a lot of fun. So getting your messages and your WhatsApp, and so you're just amazing. Thank you. None of this would have been possible without Liz, so we really appreciate it. And this is just a little token of which...
Butta la pasta. It's
you sipped
yeah,
it.
it's a gift with all types of pasta and sauces and things like that from Italy. So.
And we'll be at your house, so
Yeah.
you're the best.
So the next speaker, it's written at the bottom. You come up here. Oh, yeah. Gisela Hernandez from the University of California. Yeah. Thank you.
Okay. Good morning, everyone. I'm Isela Hernandez. I am Dr. Musafar's project manager at UC Irvine, and I'm so excited to share an update on our NIH-funded multi-center observational study in IBM, also known as INSPIRE IBM. NT5C1A antibodies are present in 60 to 70% of patients with sporadic IBM. They have a sensitivity of 70% and a specificity of 96%. They may predict a more severe phenotype, including increased dysphagia and reduced life expectancy. A prior publication by Dr. Namita Goyal found that NT5C1A positive patients were more likely to experience dysphagia and reduced FVC. Additionally, research led by Dr. James Lilleker and his team revealed that seropositive patients had, on average, an 8-year shorter life expectancy than the seronegative patients. And in order to validate and build on these findings, a larger cohort with longitudinal data is needed. So INSPIRE IBM has 4 study aims, and today's data focuses on aim 1, which is to determine whether NT5C1A antibodies mediate disease progression over 2 years, and aim 4, which is to quantify respiratory function decline in the IBM patients. INSPIRE IBM is a 2-year study with evaluations every 6 months, totaling 5 study visits, and also has an additional muscle biopsy sub-study. In regards to inclusion criteria, participants aged 40 or older were included, with the diagnosis of IBM based on NMSG 2011 criteria. And although the NMSG specifies a minimum age of 45, we did make an exception to include those aged 40 and above, and as well as disease onset within the past 10 years. We're proud to collaborate with 13 clinical sites across the US, 7 of which are our biopsy sites, which is our site, UC Irvine, as well as UCLA, Oregon Health and Science, UC Denver, Nerve and Muscle, Kansas University, and University of Miami. And here's a map of all our sites. Our assessments include physical exams, blood draws, muscle biopsies, which is optional for the sub-study, functional assessments, pulmonary function tests, as well as patient-reported outcome measures. Our central lab assessments include NT5C1A antibody testing, DNA, RNA, and PBMCs, while local labs collect standard clinical measures. We're building a robust biorepository with detailed patient data and biospecimens. PBMCs, RNA, and muscle tissue are stored at UC Irvine and Dr. Vialta's lab, while the NT5C1A antibody and DNA samples are stored at Washington University. We've partnered with leading academic and industry groups for advanced analysis, including NT5C1A antibody testing and serial tracking, single nuclear RNA sequencing, antigen mapping, and immunophenotyping. And it's been a privilege working with all of these teams. Of the 154 screened participants, 4 were screen failures, and we successfully enrolled 150 participants. Currently, we have 20 active participants, 84 have completed this study, and unfortunately, 46 have discontinued. primarily due to enrollment in clinical trials. Our muscle biopsy sub-study aims to collect 40 samples. So far, we're at 24 samples, and biopsy data is currently being analyzed, and we look forward to presenting this soon. Each of our sites had a target enrollment of 12 participants, and we'd like to give a special thank you to Johns Hopkins, Ohio State University, University of Washington, University of Pennsylvania, and University of Miami for their outstanding recruitment. And specifically, Dr. Carbonar's site at University of Miami was our final site to be activated, and they enrolled 15 participants extremely quickly. So thank you, Dr. Carbonar. Here is our baseline demographics. Our cohort is well balanced between the seropositive and seronegative groups. You can see here that 75 participants were seronegative, while 73 were seropositive, which we didn't intend on having this, but this was actually really great for us. So some of the data we're presenting today, we compared the rate of change in PFTs between the seropositive and the seronegative patients up to the 18-month time point. And although the only statistically significant difference was seen in FVC sitting, we still saw similar trends that we'll show right now on the graphs. But this table also shows the annual rate of change for each of the PFTs. So we have FVC supine, FVC sitting, MIP, and MEP. So the graphs show respiratory function trends up to 18 months, where the red line represents the seropositive cohort, the blue line represents the seronegative cohort, and the gray line represents the entire cohort. And as we just mentioned, although only the FVC sitting was found to be statistically different in the zero status of the groups, we can still see a similar trend across all PFTs. So we could see through the blue line, the seronegative group tended to have higher respiratory function in all of the PFTs, as where the seropositive cohort had lower pulmonary function results. We also analyzed functional assessments like MMT, TUG, IBM-FRS, CIFA, and PROMIS. And while no differences reached statistical significance, the annual rate of change is shown, and we believe that this is valuable data for future research. And again, this data was up to the 18-month time point, so although this does show the annual rate of change, the data up to 18th month was utilized for this. The graphs show the functional trends up to 18 months. We can see in the top left, we have MMT. Underneath, we have the PROMIS data. In the top right, we have the CIFA results. And underneath, we have the TUG. And it is worth mentioning that we did measure TUG in velocity. And our last functional assessment is the IBM-FRS. Again, this was up to the 18-month time point. And we can see the seronegative group in blue and the seropositive group in red and the entire cohort in gray. So Inspire IBM is led by Dr. Tassin Mosafar with a strong team of co-investigators, statisticians, pathologists, and coordinators. I'd like to highlight our data management team at the University of Rochester and our steering committee for their incredible support. And finally, a heartfelt thank you to our study participants, our collaborating sites, and dedicated research team. For future collaborations or questions, please feel free to contact me. And I'd also like to thank the NMSG conference for allowing me to give my talk today. my incredible PI, Dr. Mosafar, and my mentor, Marie. Thank you.
That was a great presentation. Who's got questions? But my first one I'll ask, speculate on what this data is going to mean for future prospective trials with investigational drugs. Are we going to be able to just have to go with one or the other, antibody positive or antibody negative? Or are we going to be able to mix the two?
That's an excellent question, which I do think maybe my PI, Dr. Mosafar, may be able to give a little bit more of an overall response.
No, that'd be great. Yeah, I would. Tassin, what do you think?
Yeah, so the whole purpose of this was to show if there is a difference between the two groups, and given that we've not been able to demonstrate anything, I don't think we need to stratify based on their serological status. I think we can use the combined group without consideration for the serological status.
I would be worried about that, even though you didn't reach statistical significance. I mean, on every parameter, there's.
That's correct.
Yeah, well, you can go by your p-value, or you can go by looking at the trends, and it's.
Correct.
I'd be worried that you could miss an effect of a drug if you included the non-antibody positive.
That's correct. The only statistically significant respiratory assessment was the FVC sitting, but just as Dr. Marin mentioned, we are seeing trends. Yeah. Dr. Amato also commented this during our poster session, but you're correct. Also for the functional assessments, nothing was statistically significant so far.
Mazen?
Yeah, I saw a great presentation. Thank you.
Thank you, Dr.
For the
Dimash.
FVC and FVC percent predicted, did you correct for multiple comparisons?
That's an excellent question. Right now, we did not, only because we did not, all our participants have not completed the study just yet. So we currently have 20 active participants. Our last participant out should be in January of 2026. And this was an analysis that our statistician ran for us and said we should wait until we have all the data. Thank you. Correction for comparison was utilized because of the multivariate analysis that we ran, but I don't believe we corrected for any variables such as age, sex, or anything along those lines.
Back here?
Hel-
Go ahead.
Yes, question, how positive is positive? Like, mildly positive, what do you consider positive? That's number one question for CN1A. Did you actually check CN1A at the beginning? I mean, do you have serial checking of CN1A? Those are two questions. And any correlation with clinical and serial CN1A levels?
That's an excellent question. I think I'm going to hand that one to Dr. Mosafar.
She's saying that the question is so simple that even I can answer it. So the cutoff was determined by the laboratory. So for the study, we used the Washington University Neuromuscular Laboratory for that. Additionally, we have samples that are being done in the Netherlands by Gerpruin's lab. We have some preliminary data on it, and it really depends on where the threshold is. Whether you do 2.5 times standard deviation versus 3 times standard deviation, your numbers can change based on that. So we will be presenting that data, but it's looking very similar. So if you do it at about 2.5 standard deviation, about 65% of the cohort were seropositive, and the rest was seronegative. But if you make it more stringent, you reduce the number of positives. And then we are also tracking whether patients switch from either seropositive to seronegative or seronegative to seropositive. So far, we've had 5 subjects that have switched antibodies. And then the other thing we're looking at through the Netherlands group is looking at the subtypes. So we so far, the WashU lab only looks at IgG, but we are looking at IgM and IgA antibodies, and in some patients, those dominate rather than the IgG. So I think those are some of the important.
Did you look at COO antibody? I mean, there was a nice recent paper from Werner, and if you read it, their COO patients looked like IBM, and the biopsy showed features of IBM, which made me, I always wonder whether COO antibody is not specific, that it
Dr.
occasionally
Mato,
can.
tell me, since I'm a little out of it, tell me what COO antibodies are.
Well, anti-COO is one of the myositis specific antibodies.
Oh,
it's on the
myositis.
panels. And there was a very nice paper looking at from Werner Stenzel, who's in Germany, and it's supposedly sort of like a true PM responsive, but if you looked at it, there are older patients that couldn't flex their fingers, and their biopsy showed rim vacuoles and P62 inclusions, and so I just didn't know if some patients have COO and IBM have COO, or is features of IBM can be seen in COO.
So we did not do the full myositis panel as part of it. Now, Alan Pestronk has the serum saved. We can potentially ask him to do. I was hoping that he would have done it as a side project anyway, but we don't have that data on that.
It'd be interesting with that paper to do, because
I mean, most of the patients with Q that I've seen and pathology look more necrotizing than IBM, but I mean, I definitely.
I just had one, and it was just like I scratch my head's where I'm probably going to treat it, even though I would say it looks like IBM. But with that paper out, I would do that. I think it's I love this, what you did. It's good. I mean, natural history. About Galash, we learned just what's a placebo response treatment. I mean, we can't use this necessarily, and you wonder that this is the worst case scenario of the drops. When you're in a randomized trial and you don't know, patients don't deteriorate as much. It makes me worried that an IBM declined only 1.8 or in a year and a half. That how much are you going to see? It'd be nice if we can get data from Novartis trial and maybe Apcuro that had similar, if they've had similar measurements, and compare this data.
Well, we know in the arimoclomol trial it was very similar, right, Mazen? It was the same.
I think we had a 1.8 point decline in the placebo group over the course of 20 months. Yeah.
So I don't know what Novartis was, but this is consistent with arimoclomol. So I think we know what the natural history decline is. Were you going to say something, Tassim?
Yeah, I was going to say that. So the arimoclomol group, my understanding is they did collect antibodies, but I don't think that data has been presented. And I know Apcuro for sure is also collecting information on the anti-5C1.
I was more worried
I know.
about what to use as like primary outcome measures. And does it the same? The antibody's not going to change. But does MMT, IBM FRS, that kind of stuff? What's the?
So I think that was the whole purpose, was that now at least we know what the IBM slope looks like, what the CIFA slope looks like, the TUG slope looks like, and you can choose any one of these.
Thank you.
So let me just comment. Estella, Marie, and Tassim, congratulations.
Thank
this
you.
is a huge effort, what you did. And I just think this is what we, as neuromuscular investigators, have done really well for the last 40 years, starting with the CDD group in Duchenne, and the SMA natural history, then FSH, then myotonic, now IBM. I mean, there obviously is a window in time where you can do these studies before our industry partners get involved and start developing a lot of drug options. You had 46 drop out, but that wasn't, could have. In the future, it'll be a lot worse. You couldn't even do this study. So this is really important, and this is the shoulders, the giant shoulders that industry is going to stand on as they look for a potential drug treatment for IBM. So it's hugely important. So
Thank you so
thank you
much.
very much. You wanna-
I've just
yes.
got one question which might be a bit annoying, but it's something I wonder about a lot. So IBM is a patchy disease. So the serial biopsy question, how do you kind of control for variation within an individual patient that you may not know about? And do you try to rebiopsy the same site? Obviously, it's only relevant to certain of the analyses, but I think that is an issue.
So the biopsies are not serial. It's a one point.
I thought you did 3 biopsies.
No, just
It
one.
could be taken at either of those study visits, but it's
So,
only one.
Okay.
We didn't mandate
Okay.
that it has to be only at baseline. We could do it anything. And the purpose there again was to see, and there were some reports initially by Andy Mammoth's group that there may be muscle pathology differences between seropositive and seronegative, and that's essentially what we are trying to answer here. But these are a single time point biopsy, but it could be done at either baselines, month 12 or month 24.
But I always think, I don't know what other people think, there's a concern about the histopathology will vary in different parts of the same patient, and I'm not sure exactly what you do about that.
Yeah.
But you have to be careful about interpreting. If you do serial ones, you have to be careful about interpreting what that means.
I completely agree with that.
Yeah, so I thought it was a great session, great talk. Thank you very much. And so we'll hand over now to the moderators for the next session, Young Investigators. Thank you.
Thank you, everyone.
So hi, I'm Karen Sutherland from Newcastle, and it's my pleasure to introduce Dr. Murva Assad from University College London and to talk about MAP, MRI as a biomarker in periodic paralysis, a prospective longitudinal pilot study in periodic paralysis.
Thank you. Thanks for the introduction. So my name is Marwa. I'm a clinical research fellow with the Channelopathy Service at Queen's Square. Thank you for the opportunity to present our team's work on developing MRI as a biomarker in periodic paralysis. So for context, the periodic paralyses are rare inherited conditions consisting of hypo and hyperkalemic periodic paralysis and Anderson-Towle syndrome. They're caused by mutations in ion channel genes, namely CACNA1S, SCN4A, and KCNJ2. These disorders are quite heterogeneous, ranging from episodes of weakness provoked by specific triggers to progressive myopathy that can be quite disabling. And while we have some symptomatic treatments, we don't have any disease-modifying therapies to prevent this progression. And this is, of course, a real priority for our patients. But when we talk about novel therapies or disease-modifying therapies in clinical trials, one very important aspect of this is, of course, having reliable outcome measures or biomarkers. And because these disorders are so variable, clinical scores aren't really suitable for these purposes, so there is a real need for a biomarker, an objective biomarker, and we propose MRI as this much-needed biomarker. So we already have some cross-sectional data from our department showing, this cross-sectional data showing significant intramuscular fat replacement, which you can see in the image there, and also stir hyperintensity being a prominent feature. But longitudinal studies are very few. Our colleagues in Copenhagen have presented some longitudinal data in one genotype, in CACNA1S, and that has shown this progressive fat replacement across multiple muscles in patients both with and without attacks. So these studies are encouraging and place MRI at the center of the search for a non-invasive biomarker. So we were fortunate enough last year to get some funding from the NMSG Shark Tank Award for a pilot study, incorporating some of our aims and some of the key features we identified as a need. So we felt we needed more longitudinal evaluation of MRI changes, the inclusion of quantitative assessments, particularly using Dixon or T2 and T2 mapping, these more physics-based MRI biomarkers, and exploring, of particular interest to us, newer advanced diffusion sequences. Now, unfortunately, because of unforeseen damages to our MRI machine, we've had some delay in collecting data for the prospective part of our study, but I'm going to present some of the analysis of our current longitudinal MRI data and some of the key findings of that today. So for our analysis, we included adult patients with genetically confirmed periodic paralysis who were consented to a cohort study and who had both baseline and interval MRI scans of the lower limb muscles. The images were acquired on a 3T MRI machine using more routine imaging protocols, such as axial T1 weighted and stir images of the bilateral thighs and calves. We use these semi-quantitative scores to score the muscles in fat replacement using the modified Mercury score, muscle atrophy using this 4-point scale, and stir intensity using this 3-point scale. We took the mean score of left and right muscles and used that for the analysis. And the images were scored by an experienced neuromuscular radiologist. So our study included 15 patients in total. That included the majority had hypokalemic periodic paralysis, but we also included 3 with sodium channel mutations and 5 with potassium channel mutations. 6 of our patients were female. The average age at baseline was 38, and the mean scan interval was 4.5 years. Of note here, the majority of 12 out of the 15 patients had some degree of fixed weakness at baseline. And the majority also had attacks of weakness within the last year. 2 were using gait aids at baseline, and all patients were on some form of symptomatic treatment. So before going into the MRI results, I just wanted to highlight the clinical course of our patients between scans. So the median modified Rankin scale, a measure of disability, progressed from 2 to 3, so a slight to a moderate disability. And just to highlight the heterogeneity of periodic paralysis, 6 patients progressed, 7 remained stable, and 2 actually improved. So again, perhaps highlighting that these broader clinical scores are not as useful, but also highlighting the degree of disability in this cohort. We went from 2 gait users to 6, so 4 patients newly required gait aids in that time. 2 of the previous 3 patients who didn't have fixed weakness developed fixed weakness, and 2 patients' phenotype also changed from episodic weakness to progressive fixed weakness. In terms of the MRI, this was abnormal in either one or more domain in all of our patients, and I'll go into that now in more detail. So this heat map just highlights the baseline fat replacement across different muscles in all of the patients, divided by thigh into the thigh and the calf. So you can see multiple muscles are affected, and perhaps the adductor magnus and the medial gastroc emerge as the most affected at baseline. So the fat replacement did progress. At baseline, about 43% of muscles were affected, and that's in 12 out of the 15 patients. The mean Mercury score was 0.79, and you can see from the graph there that there was a small but measurable increase in the mean Mercury score in all of the muscles, particularly, again, the adductor magnus and the medial gastroc. The mean change across all the muscles was 0.17, so again, small but measurable, and in all the muscles. And this is just some example images in a patient with hypokalemic periodic paralysis. So the top image is the degree of fat replacement in the thigh muscles at age 51, and then you can see by age 58 there's almost complete fat replacement of some of these muscles in the adductor magnus, the vastus lateralis, and the posterior compartment. Atrophy was less of a feature. At baseline, it was only present in 7.7% of muscles, and the mean score was quite small. While there was an increase in half the patients at follow-up, it was quite a small increase. And as you would expect, it did correlate with the mercury score in specific muscles. The changes in stir intensity were interesting. So at baseline, there was stir hyperintensity noted in about 20% of muscles and in nearly all individuals. However, at follow-up, it was either unchanged in the majority of patients, increased in about a quarter of patients, but also decreased in some. And that decrease is interesting. We couldn't specifically say that it was attributed to medication changes, as all patients had some change in their medication over the course of the study. We can, however, say that the stir was more of a feature in the calf versus the thigh, both at baseline and in terms of increase or change. We would sort of interpret the genotype signals cautiously because the numbers were small, but it seemed at baseline the sodium channel mutations had more fat replacement, and that the Anderson-Towle patients, or the patients with potassium channel mutations, didn't really have as much structural change in the lower limb muscles, and perhaps MRI of the legs is not as useful in this cohort. So to summarize our key findings, so firstly, clinically, there was progressive functional decline in this quite young cohort, as evidenced by the progression to fixed weakness, the increase in the MRS score, and the increased reliance on gait aids. No patient had a completely normal MRI at baseline. We did see fat progression. This was measurable, but it was quite modest, and so possibly T1 weighted images are not the best for shorter trials, or would have low sensitivity for that. We did see subclinical MRI changes, however, so that is useful, and it also highlights to us that, again, the broader clinical scores are not as useful. The MRS didn't necessarily change, but there was more fat replacement. There might be a temptation to cut MRI times and include, perhaps just the thighs, but we found both were actually useful to include in studies going forward. Again, we highlight that in Anderson-Towle syndrome, lower limb MRI is not as useful, and perhaps paraspinal muscle imaging or pelvic muscle imaging would be more useful in this cohort. The stir intensity is interesting. It was present in nearly all patients, more so in the calf than the thigh, and we did see improvement in some. So the longitudinal value might be limited, but responsiveness to treatment would be interesting to explore further. There were obviously some limitations to this aspect of the study. It was retrospective, and the cohort was small and heterogeneous, and we had no healthy controls. So for our prospective study, we're going to take some of these learnings into account, and essentially we need more sensitive quantitative imaging and the inclusion of healthy controls. So I'll just briefly go through our plans for the next phase. So we heard yesterday from Dr. John Thornton about these physics-based MRI biomarkers, and he spoke about T2 relaxometry, which we do plan to include. But I'll talk a little bit about the diffusion aspect, which is increasing in use in the neuromuscular field, already used quite a lot in brain imaging. So it is sensitive to changes in myofibril permeability, and that's been shown in Duchenne animal models. And our collaborator, Dr. Amy McDowell, has also demonstrated that children with DMD, when using longer diffusion times, you can detect the differences between healthy and diseased tissue. So for our prospective study, we want to look at the feasibility and reproducibility of these advanced diffusion sequences in PP. We plan to recruit 5 patients who we've already identified and include 3 healthy controls. We plan to scan them at baseline 6 months and 12 months. They'll receive standard therapies, and we'll have corresponding clinical assessments at each clinical visit. And again, because we're moving to a new scanner, this MRI protocol does need to be finalized, but this is what we plan to include. So more standard sequences at a shorter coverage in the thigh and the calf, a larger field of view using a 3D Dixon with our 58 centimeter coil, and then including T2 mapping and the standard and longer diffusion times. We're going to start with 4 diffusion times in this pilot study to identify the optimal diffusion time, and then hopefully go forward with that with a shorter scan time in the next phase of the study. So we're excited to start this over the next month. And I'd just like to thank my supervisors and collaborators, and hopefully we'll be back next year.
Thank you. I think we've got a question.
Thank you for this excellent presentation. From a quantitative standpoint, I was just looking at my copies of the slides of Dr. Thornton earlier. He mentions about measuring muscle cross-sectional area as one of the outcomes to look for as a biomarker of disease activity. Did you already measure that? I missed it somehow, or do you plan to measure it?
The cross-sectional area
Of viable muscle?
of viable muscle. I mean, I suppose we sort of looked at it in the atrophy, where the area would be reduced of the viable muscle. It wasn't as useful, but of course a lot of it was replaced by fat.
Yeah. I think there's a bit of more quantitative way of measuring that that he was alluding to.
Yeah.
Thank you.
Yeah.
I think we've got another question at the front.
I wanted to ask you as well, did you have any variable of symptom onset or age at diagnosis? It looks like your lower end of patient acquisition was in the 20s, but I was wondering if there's any difference in kids that are diagnosed at maybe 10 and number of episodes that they've had in correlation with the MRI findings as opposed to somebody who has symptom onset maybe a bit later in life.
Yes, yeah. So this was, the age range was kind of vast in this group. The average age was around 38, and the lower end was in the 20s. We're just an adult service, so we didn't start the data collection from childhood. But on average, the onset of fat replacement is usually past around the age of 40. So we wouldn't expect it as much in the lower group. And I think, I don't have the slide.
Can we get the slide back up?
I think if we go back to the heat map, you can see it's not as much of a feature in the lower age groups, so kind of in the 20s, less so. The older calcium channel patients certainly have a lot of fat replacement, and then you can see in the middle there at age 38, a sodium channel patient, so at a younger age they may start to develop fat replacement. But our plans for the diffusion and the Dixon imaging would be that it might be more sensitive to detecting some of those changes earlier.
It's just interesting that some of the kids who have multiple episodes will have weakness as well. I mean, they will have findings of weakness when they've had multiple episodes. So I was just interested in the sort of the rate of change and whether or not it would be something that we would be able to see as well.
Yeah, that would be great for the prospective study. So our plan with that is to scan patients at a younger age before a lot of this fat replacement, because that will confound the diffusion and the quantitative T2 imaging. So I guess we might know from that how young that fat replacement might start and whether lots of attacks at a younger age change that rate.
I think Valeria.
Just a quick thing, maybe I missed it. The treatment does not seem to have changed anything. I mean, all these patients were on treatment, right?
Yeah, they were all on some kind of treatment, so either acetazolamide or diclofenac or just potassium-altering treatments, and it doesn't seem to have affected that progression.
Yeah.
And these numbers are small, but...
Because the concept of the number of episodes giving sort of theoretically being responsible for the permanent myopathy, and then it's related to the treatment, but then...
Yeah, I mean, that theory, I guess it doesn't necessarily hold up. I think others have shown that despite no attacks, the myopathy might progress.
Yeah,
so,
the same with treatment. It doesn't seem to...
Yes, yeah. So they're not disease modifying, I guess, these treatments. Yeah.
Brilliant. I think last question at the back.
Thanks. Nice talk. Have you some hypothesis about the fat replacement in mastocyanulopathies? Second question is about histopathological data. Have you some muscle biopsies of this replaced muscle? And if you check some association with the severity of fat replacement and clinical data, like the number of attacks during the life of patients or some other clinical data.
So on biopsy, some of these patients show vacuoles, so they show almost like a vacuolar myopathy. But the reason for this change from episodic weakness to progressive myopathy is not understood. We don't know why exactly that happens. And what was the other aspect of your question? The amount of attacks. Yeah, so we've said that kind of similarly that it doesn't seem that a lack of attacks will prevent progressive myopathy. We do know, just sort of anecdotally from our cohort, that a really severe, prolonged attack, patients recover less well from. So maybe the severity and duration of the attack itself might have an impact, but some patients have frequent attacks, some have very few attacks, but still progress to myopathy. So it doesn't seem to correlate exactly.
Mike.
Yeah, I was just going to make the point that I think the jury is really out about this relationship between attack frequency and severity. And when you sort of think about it, why should it? You know, because like the channels are dysfunctioning the whole time. I think the main message is these patients are developing a progressive fixed myopathy that I don't think our drugs really intervene on significantly. And my view is ultimately genetic manipulation therapies to alter the mutations will actually be the more likely way to prevent the myopathy. Because when you look at the data on attack frequency and severity, it's not really solid. So I think drugs are useful clinically for helping patients have less attacks, where the patients have a lot of attacks. But I don't say to patients, and this will make you less likely to have a myopathy. I think that's open comment really rather than the question.
Thank you.
Great,
thank you.
much.
Thanks very much.
So just a quick housekeeping announcement before the next presentation. So there's going to be an addition to the agenda. Last year's Shark Tank winner will be presenting after the next speaker. So we're going to welcome Dr. Massey from Yale University to talk about IgA autoantibodies as a novel mechanism in anti-MuSK myasthenia.
So thank you, and good morning to everybody. I'm very excited to present our research on MuSK myasthenia gravis. So these are my disclosures. So it is well known that patients with MuSK myasthenia harbor antibodies of the IgG class that target MuSK, the muscle-specific tyrosine kinase on the muscle end plate. MuSK can interact with other two proteins, LRP4 and neural agrin, which is released from the motor nerve terminal. And this interaction will promote MuSK dimerization and phosphorylation of the kinase domain, which will trigger a complex cascade that will eventually determine and promote the clustering of acetylcholine receptor on the muscle end plate. And we can recapitulate this mechanism in vitro by incubating myotubes with agrin that will trigger the clustering of acetylcholine receptor on the muscle surface. Now, one of the interesting and fascinating features of MuSK myasthenia is that the majority of MuSK IgG antibodies are IgG4. And IgG4 antibodies are able, because of specific amino acid residues in the Fc constant region, to exchange half molecules with unrelated IgG4. So the resulting antibody is bispecific. That means that MuSK IgG4 antibodies will have a single arm that will bind to MuSK. And it is well known that these antibodies are pathogenic. They can bind MuSK with a single arm, preventing the interaction of MuSK with LRP4. And because of the blocking of this interaction, MuSK is not dimerized anymore, and AChR receptors are not clustered. And again, we can test this model in vitro by incubating myotubes with agrin, and with the Fab portion, a single arm of a patient-derived MuSK-specific IgG4 antibody that will result into loss of AChR clusters. Now, Beyond IgG, there's evidence that other antibodies can contribute to immune mechanisms, and one of them is IgA antibodies. IgA antibodies represent the second most abundant immunoglobulin in serum, and there is growing evidence suggesting that these antibodies can contribute to immune mechanism in neurologic disease. And here, I wanted to highlight a few examples ranging from autoimmune encephalitis to myelin oligodendrocyte glycoprotein antibody disorder to multiple sclerosis. And so this is a photo of me last year in Terrytown, and I was talking to the sharks, and I was sharing some preliminary data. Basically, I was telling them that I performed a preliminary experiment by testing patient sera using a cell-based assay. So basically, incubating sera with mask-expressing cells. And what I found is that a subset of patients with myasthenia gravis harbor mask-specific IgA antibodies. And I was giving my elevator pitch, trying to persuade the sharks that it could be a good idea to study masked IgA antibodies at the molecular level in order to provide novel insights into disease mechanisms. And now here I am several experiments later. So first of all, we were able to confirm that masked IgA antibodies are found in a subset of patients with masked myasthenia. These are the preliminary results, but we also tested a second independent cohort. Now, all patients harboring masked IgA antibodies also harbored masked IgG antibodies, but we didn't find a correlation between the levels of the two antibody classes. Now, these are representative fluorescence microscopy images. This is a cell-based assay result. So in green, you see mask-expressing cells, but in this case, you're looking at the control, so you don't see any IgG or IgA binding. This is a patient with masked myasthenia gravis harboring IgG antibodies, and on the right, you see a patient harboring both masked IgG in blue and IgA antibodies in red that colocalize very well with mask-expressing cells. Now, among all patients with masked IgA antibodies, one was quite interesting because we were able to collect longitudinal samples over 12 years of disease activity. So you see in blue, masked IgG levels, in red, masked IgA levels. And it is well known that rituximab works quite well in myasthenia. And we observed that both IgG and IgA levels diminished following rituximab administration, which is represented by the orange dashed line. However, we found a differential response to rituximab. Basically, we found that the IgG levels diminished at a greater extent compared to IgA levels, suggesting that mask-specific IgA B cells may be more resistant to CD20 depletion. And in order to dissect the molecular features of these antibodies, we decided to look at a single antibody at a time, producing patient-derived recombinant monoclonal antibodies. So basically, we isolated B cells from patients. We selected the cells that were positive for masked IgG, and we cloned recombinant monoclonal antibodies. Now, this process is quite challenging given the exceptional rarity of mask-specific B cells in circulation. And just to give you an idea, after screening more than 130,000 B cells in the periphery, only 6 B cells were found to express mask-specific IgA. We were able to clone 3 recombinant monoclonal antibodies, and the fourth column indicate the number of amino acid replacement in the variable region, indicating that these antibodies are affinity matured. These antibodies bound well to mask using both flow cytometry and microscopy, and these are their relative binding strength curves. And interestingly, these antibodies derived from 3 distinct patients could bind to mask and specifically targeted distinct mask domains, like the IgLEC1 domain, the IgLEC2, and the frizzle. Furthermore, these antibodies could also compete with IgG4 antibodies for binding, suggesting that IgA and IgG4 antibodies could bind to the same or partially overlapping epitopes. So I've shown you that these antibodies derived from patients can bind to mask and are of the IgA class. Now, because each antibody is basically like a fingerprint of a B cell, given that we have the sequence of the antibody, we can look at clonally related B cells. Basically, these are B cells that share a common ancestor with a B cell of interest. And we found a large family of B cells expressing masked IgA antibodies in a patient. Now, these B cells were found in different time points and again resisted several administration of CD20 depletion. Now, these antibodies bind very well to MuSK, but what is the effect of these antibodies on the neuromuscular junction? And so, as I've shown before, the MuSK IgG4 antibodies can bind MuSK with a single arm. But MuSK IgG antibodies are bivalent, so they can force dimerize MuSK, inducing phosphorylation and promoting AChR clustering. And for the remaining of these experiments, I will focus on two antibodies because they showed cross-reactivity to both human and mouse MuSK. We were able to confirm indeed that the MuSK IgG antibodies are able to induce, number one, MuSK phosphorylation. They can also promote AChR clustering, but in the presence of agrin, they reduce agrin-dependent clustering mechanism. Now, we also performed another experiment. This is an in vitro disease model in which we incubating myotubes with a patient-derived MuSK IgG4 antibody. And we also added a MuSK IgA antibody, given that MuSK IgA and IgG antibodies can coexist in patients. So these are the results of this experiment. We know that when we add agrin, we promote clustering of the receptor. When we add a MuSK IgG4 antibody, clusters are basically gone. However, the addition of MuSK IgG antibodies will partially rescue cluster formation. So it seems that in this specific experiment, the antibodies are compensating. They're triggering a compensatory and protective effect. However, what I didn't mention is that in serum, MuSK IgG antibodies can be polyclonal. And this is an example of a patient harboring antibodies targeting to two different domains. And so I was curious in the last experiments to combine MuSK IgG antibodies together. So in this experiment, you are looking at myotubes that are incubated with two MuSK IgG antibodies, MU1 in orange and MU3 in blue. These antibodies are able to promote clustering. However, and this was quite intriguing and also counterintuitive, when we combine the antibodies together, we see a profound loss of AChR clusters. And even in the presence of agrin, the effect was quite striking. Now, this effect was not simply due to the addition of more IgA, because combining one MuSK IgA with 8-18C5, which is a MOG-specific IgA antibody, we didn't see the same result. So basically, it seems that the addition of two antibodies can exert highly pathogenic effects in vitro. So I've shown you that MuSK IgG antibodies have pathogenic capacity in vitro. So the next question is, what about their pathogenic capacity in vivo? And so we wanted to investigate this further, and this is why we performed a passive immunization model. So basically, we injected MuSK IgG antibodies in a group of mice. The antibodies were combined together to mimic serum polyclonality. And we compared the effect of MuSK IgA versus KLH IgA, which is an innocuous antibody. We injected antibodies daily. We monitored body weight as well as muscle strength, so grip strength and hanging test. So these are the results of this experiment. In black, you're going to see the mice receiving MuSK IgA. In red, the mice receiving a control antibody. So over time, the mice receiving MuSK IgA lost body weight. We also observed a reduction in grip strength. And they also performed poorly in the hanging test. And these are two images of, on the left, one mouse receiving a KLH antibody. On the right, a mouse receiving a MuSK IgA antibody. And you can observe the difficulties that the mouse is experiencing on the hanging test. We also wanted to look at the neuromuscular junction. And so after the experiment, we confirmed that the IgG antibodies could indeed bind to the neuromuscular junction. In red, you see the deposition of MuSK IgA colocalizing with the bungarotoxin signal. But what about the morphology of the neuromuscular junction? And so on the left, you can see pretzel-like shapes. Those are the neuromuscular junction of a healthy mouse. On the right, a mouse receiving MuSK IgA antibodies, and you can see a profound structural disruption of the acetylcholine receptor, indicating that these antibodies are able to induce a myasthenic phenotype in mice, and they also alter the structure of the NMSG in vivo. So to conclude, we identify a novel pathogenic isotype in a subset of patients with masked myasthenia. The masked IgA cells also exhibited remarkable resistance to the CD20 depletion, possibly suggesting that these B cells can be a cellular source that can trigger masked autoimmunity. Now, when we performed experiments with single antibodies or combination, we found striking differences. Single antibodies could behave as masked agonists, inducing clustering of the acetylcholine receptor, but the combination of two antibodies was highly pathogenic. Why? This is like a speculative working model. We know that agrin can activate the mask pathway. In masked myasthenia, what happens is that the masked IgG4 antibody will block masked LRP4 interaction. If we look at masked IgA instead, single monoclonal species will force dimerize mask in a highly ordered way, promoting clustering, but we speculate that the combination of multiple antibodies binding to multiple domains of mask will trigger a dysfunctional cross-linking, and the stability of clusters on the muscle end plate will be diminished. And so there are obviously outstanding questions. Number one, can these IgA cells class switch and produce IgG4 antibodies? What is the efficacy of IgG-targeted treatments on patients with masked IgA antibodies? And finally, given the isolation of masked IgA antibodies, are mucosal triggers responsible for the autoimmune response against mask? And so this is something that I also mentioned last year. We are interested in evaluating whether the masked IgA antibodies and the masked IgG4 antibodies can bind to bacteria. And we are performing some experiments. And I'm also happy to continue this experiment through a recent grant that I received. Finally, I'd like to thank my PI, Professor Kevin O'Connor, all the lab members, our collaborators, the neuromuscular study group for the support, and last but not least, I'd like to extend my best wishes to the contestants of the Shark Tank of this year. Thank you.
Questions? I have one. So it looks like in your cohort, there was just a few patients that had the concurrent antibodies. Is that right?
So all patients that are positive for masked IgA were also positive for masked IgG.
Yeah. Did you or are you planning to look at the clinical characteristics of those patients?
Yeah, we did some preliminary analysis. Now, given the limited number of patients, we didn't observe striking differences when we compared the masked IgA positive and masked IgG. Masked IgA positive and masked IgA negative patients. So these patients didn't differ if we think of like clinical onset, like disease duration, and response to treatment. However, one interesting observation is that in one patient, the masked IgA response was persistent over time, and this patient was dependent on multiple CD20 administration, possibly suggesting that the presence of masked IgA could signal a chronic dependence on CD20 depletion. Obviously, this is a hypothesis that needs to be confirmed using more samples. Thank you.
Hi, Jane Vito. Great talk. I had a question about the binding sites of the IgA anti-mask antibodies. Did you look at the specific binding sites for them? Because my understanding of like Steve Burden's work is that if you bind to the Frizzle domain of the musk receptor, is that promotes the phosphorylation and it acts as a musk agonist, whereas if you bind to other regions of the musk protein, that is inhibitory because it blocks the binding site of LRP4.
Yeah, thank you for this question. So I would say that what we know about the mask LRP4 interaction is that it's a complex interaction. So it is well known that a mask antibody that target the Frizzle domain can induce mask phosphorylation, and actually it's been tested like therapeutically. We know that it works well in a model of congenital myasthenia. What we found, however, is that based on our experiments, is that any antibodies in this case IgA antibodies, independent of the binding site, they will promote mask phosphorylation because these antibodies are bivalent, so they will force dimerize mask. Now, obviously, the epitopes are important because we know the Ig-like-1 domain also interacts with LRP4. There's, however, some growing evidence suggesting that actually mask antibodies targeting the Frizzle domain can be pathogenic. There's a recent paper published in JCI by a French group. In that paper, they were looking at the polyclonal mixture of antibodies targeting the Frizzle domain. So it seems, and this actually aligns with our experiments, that while single antibodies can be protective, for example, if they bind to the Frizzle domain, a polyclonal mixture will be detrimental. There's also some evidence that the Frizzle domain can also interact with LRP4 through a distinct domain. So it's possible that the antibodies will also, if they're polyclonal, they will bind to other epitopes, blocking the interaction between MuSK and LRP4.
So I guess you're explaining why there might be some patients with a MuSK that don't improve with Rituxan might have pathogenic IgA antibodies. But would there be a difference with anti-CD19 therapy, or is that going to be the same with the anti-CD20?
So we haven't performed these experiments yet, but we're interested in looking at the phenotype of the B cells that produce MuSK Ig antibodies. So we found that those cells were resistant to the CD20 depletion. And so we have some hypotheses. One is that those B cells are found in tissues, and some treatments cannot target those tissues. I'm thinking about the gut mucosa, for example. It's also possible, however, that the B cells that express MuSK Ig antibodies lack CD20 expression. So an important idea, actually, I agree, is that other treatments and other targets like CD19 can also target those cells and eradicate them. But this is something that we have to investigate further.
Great. Thank you so much.
Thank you.
So we'd like to welcome Dr. Giorgio Carati from Rome to present her talk on the Shark Tank from last year.
Hello. Just a disclaimer, this is not from last year. Oh. Actually, I won the Shark Tank in 2022, but last year I had COVID, and long COVID, I was very sick, so I was forbidden to travel and give you an update. So thank you very much. If I can have the slides. Yes, thank you. So today I'm here to introduce you to our preliminary data that are even better than last year, so I'm kind of happy of that. And our project was on machine learning to predict disease progression in spinal muscular atrophy. So everything actually started several years ago with our first paper on the use of machine learning to predict SMA in natural history. And in this paper, we were able to prove that it was possible to use this approach at least to understand and predict disease progression on the Hammersmith Functional Motor Scale Expanded, that is an outcome measure that we often use with SMA type 2. Our research question, after all the disease-modifying treatments and the enormous improvements that we see in these patients, was to really understand how these patients now respond over time, because as clinicians, we do see a lot of variability in treatment response in these patients. So we were really puzzled to discover which variables were the one leading the response of treatment over time. So our aim, that is why I won that Shark Tank in 2022, was to create a predictive algorithm with the use of AI, integrating clinical data, patient-reported outcome measures, and biological markers, specifically those one listed in this slide, so they're circular RNAs, that were supposed to help us in defining these disease trajectories. Unfortunately, the biomarker part with the circular RNAs was not successful as a predictor of disease progression, so we needed to delete that from the model. But what we did was to try to apply a combination of approaches that encompass descriptive statistics to AI predictive modeling. And what we did was to study each outcome measure that we have available for these patients that are in the specific HMFSC, as I said before, the revised upper limb measure, and the CHOP intent for infants, and try to understand with every linear mixed model how the scales were behaving for each phenotype. And at the end of this, that is part more of a descriptive approach, we used a latent predictive model that was able to capture overall disease progression as a combined outcome on a motor level. So the model was built as classically, you also have listened yesterday to the presentation of AI, but classically the database is split in 80% of training set and 20% of testing set, with the variables that were pre-specified in our analysis, so that were age, treatment visit, SMA type, SMN2 copy number, and then a number of clinical variables like ventilatory status, nutritional status, scoliosis, the Hammersmith score for the Hammersmith prediction, of course, and the treatment interaction with age. So what we were able to achieve as preliminary results are two kind of prediction. So we have the possibility to have a full prediction in which the score is predicted on the basis of the previous visit. And then we have a chain prediction. And the chain prediction is really that unsupervised approach that everyone was speaking about yesterday. So being able to predict over time without having the information time by time. So with only the characteristic at baseline. So these are some examples. So if you see the blue line is the one representing the real data from this particular patient. This is an individual with SMA type 2. Then you have the red line, that is the full prediction, and then you have the orange line, that is the chain prediction. And what we are predicting here is the Hammersmith over time. And if you remember what I just said, the full prediction is predicted through the previous values that are all the variables that I explained before, while the chain prediction was drawn with only the data from baseline. And you can see that the model, of course, has an uncertainty, especially for the chain prediction, but the trend overall is very well good predicted. And this is another example of another patient, and you can see how the tree line goes overall together into the prediction. This will be, in our opinion, useful to understand even with just the information at baseline, how that patient will progress over time, and then the influence of the treatment of these patients. As a result of these even difficult models to understand, even for myself, the statistical group that made this analysis also tried to combine a combined motor outcome to predict progression of disease progression in these patients. And what they were able to do was really to identify in an unsupervised way, so nobody told the model how many classes we wanted, but the model was able to identify 3 classes of responder in our data set. For example, in SMA type 2, using this model, this latent model, the best model that was chosen by the algorithm to predict the disease progression in this patient was exactly a 3 latent class model. And applying these classes to these patients, we can see that, for example, on the Hammersmith, the model is able to identify, without seeing the data before, patients that are in the red, so in the first class, that we will call super responder for a clinician-friendly approach. Then the blue ones that were the responder, presumably, and then the white that are the known or less responder. As you can see, there is kind of an uncertainty in this, specific piece of the graph. And here we are trying to refine the model and to understand why the algorithm is not able to correctly identify these patients. But overall, the concordance between the classes and the progression over time was 70%. So I just wanted to thank you for letting me presenting you this data, and I hope next year we will have even more better data. Thank you.
Show of hands for any questions.
So I'm just curious, as somebody who's naive, when you're making a prediction model like this, how big a team does it take to make the computer programming portion of this? And how do you choose a tool? How do you pick which, how the tool is going to analyze the data for you?
Yes. Well, for sure, it's not just me, as I said. I'm a physical therapist by background. We have a team of biologists that were in charge of the biomarkers, of course, but we have a facility inside our hospital that is specialized in artificial intelligence. So we have the smart guys making all the work. In order to model something like this, you have to have a lot of data points. And in this model, for example, we have 3,000 visits for SMA patients. And so this is why we were able to have a preliminary model. Of course, this will ever be a proof of concept unless we incorporate other data, and this is the next step that we are going to do. We are going to apply for different grants and try to expand that on a national level, and then having an external validation on an international data group. So, yeah.
Can I just make a quick comment? So I think this is, again, stressing the importance of collecting lots of data over time with good quality, because the idea of the model, other than the smart guys, you have to go to the smart guys with the variables that have been identified in the natural history studies that you already know are the predictive ones. What AI is doing, if I understand correctly, Georgia, because I'm also not one of those, you put the things together and then that's the output. And of course, it really depends on what you know before and the strength of the longitudinal observational data and the quality of the data. So I think that's the message for using AI. Otherwise,
yes,
it's not useful.
Yes, modeling is like a multidisciplinary team. You have to have all the pieces, because otherwise, the statistician will always think about statistics and, that's everything, and the clinician just the clinic part. So you have to have every bit. Thank you very much.
I think we'll now hand over to the Shark Tank and to James.
So we might just need a couple of minutes to get ready. I don't know if the Sharks want to come down to the front and collect their score sheets and their costume. And there are people sitting right at the back. There is space at the front if you want to come forward. And the tables have these QR codes on to do some voting, so that might actually help if there aren't any QR codes at the back. Is that everyone?
I can't see very much.
Okay. Oh, and this is just a very cheeky advert from me. We're advertising at the moment for a neuromuscular research fellow in Manchester to come and join us. So if anyone wants to come and work in sunny Manchester, then please give me a shout and scan that QR code. It's QR code overload here. So, are we all ready? We're all ready. So I'm going to take these off if that's okay, because I can't see what I'm doing. So yeah, I'm James, neurologist from Manchester. It's my pleasure to chair this next session with the help of the Sharks here. And as you've seen from the last few talks, I think this has really grown from its inception, I think, around COVID era into something now which I think is funding lots of really exciting projects and most importantly, I think, really developing and supporting our young investigators, and that's really the key aim. And whilst there is a prize here, so the winner gets $10,000, and there's at least one winner, you'll notice that some years there's more. Sometimes Rick says, oh, look, we've got enough money, we'll fund a couple more. So there'll be at least one winner. But the money isn't everything, to be honest. All of the speakers will have the opportunity for this experience, the feedback from the panel, the feedback from you as an audience, and also I provide them with a sort of written summary as well afterwards, and they invited to have a mentor session with a member of the NMSG as well. So this really is about supporting the neuromuscular experts and specialists of the future, and I think that really is the most important thing. So here are our previous winners. There's a lot now. This writing's quite small, but this really shows you how I think how beneficial this has been. If you look at Vino here back in 2020, this study's now been published in Lancet Neurology. Katie's now working with me in Manchester as a consultant colleague, and just down the list here, you heard Merva, you heard Gianvito today. Did we hear? Who did we heard another talk today? Where is this on the list here? Did we miss that? Maybe we missed that. I'm sorry, but we had an extra Shark Tank speaker today. So, without further ado, then let's move on. So we have our sharks. I'll ask each shark to just introduce themselves and say where they're from. And then we shall move on. So should we start with Nicoletta on the end there?
Yeah. Hi, my name is Nicoletta Nicolenko. I'm a senior clinical research fellow at the University College London Hospitals.
Gordon Smith from Virginia Commonwealth University.
Jasper Morrow, neurologist at Queen Square. And just to the presenters, although a group of sharks is called a shiver of sharks, don't be scared, we won't bite.
Usually.
Hi, Carleen Muil. I'm from Radboud University Medical Center in the Netherlands. I'm a neurologist, and you do need to be a little bit afraid maybe my teeth are sharpened.
So one of the exciting developments this year, and I think this is Valeria's idea, but Valeria has all of the best ideas, is to get a bit more audience participation. So you will be invited to ask questions for each talk, but also now there's the opportunity to give some more structured feedback via the QR codes, which will be shown on the screen, but also on the desks in front of you. So you'll sort of see that each speaker has an individual QR code. You can vote once for each person, and you can only vote whilst that person is on the stage. So you have to sort of be listening and ready and good to go. But as that speaker's talk comes to an end and you listen to their response to questions, then scan your QR code and vote, and that your input then will be taken into account when the sharks are making their deliberations and deciding who's the ultimate winner this year. So we'll look forward to seeing what you say. And the winners are going to be announced at 12:15. I think that's right. James, tell them that the QR code will be on the screen. The QR code will be on the screen. We've got copies here. So here's the first QR code. But these are on your desk. So yeah, you'll find it there. So yeah, I'm sure you know all how to work a QR code. I hope so. So are we all ready? Yeah. Are we all ready for the first talk? So if we can have the slides up for Silvia Faso, Catholic University of the Sacred Heart, investigating cancer-triggered autoimmunity in myasthenia gravis. And just to mention, so the speakers get about 5 minutes, 3 slides. It's really a bit of an elevator pitch. Most of the interaction for about 15 minutes will be with the sharks. and then we go through the 4 speakers. So thank you very much. Sylvia, over to you.
Good morning, everyone. Good morning, Sharks. I'm Silvia Falso, and I'm a neurologist at Catholic University in Rome. I'm a PhD student, and I'm really excited today to present my project investigating cancer-triggered autoimmunity in mask myasthenia gravis. As Gianvito said, mask myasthenia gravis is an autoimmune disease characterized by the presence of autoantibodies directed against mask protein that block AChR clustering, resulting in muscle fatigability. MASK is a receptor tyrosine kinase, and to date, the mechanism leading to the production of masked antibodies are still not clear. Masked antibodies mostly belong to IgG4 subclass, and IgG4 is an anti-inflammatory antibody typically associated with chronic exposure to an antigen, and in the last years, growing evidence suggests that IgG4 may contribute to cancer immune escape. And IgG4 responses have been reported in patients with different types of cancer. And so we decided to perform immunohistochemistry to assess mask expression on 2 tumor specimens from 2 patients that developed mask myasthenia gravis after cancer diagnosis, one primary mediastinal B-cell lymphoma and one endometrial clear cell carcinoma. And you can observe that both tumors were strongly positive for mask in the nuclei, so at the nuclear level. And in figure A and B, you can observe the primary mediastinal B-cell lymphoma, and in figure C, the endometrial clear cell carcinoma. Then we decided to perform immunohistochemistry on other 2 PMBCL and other 2 endometrial clear cell carcinoma from patients without myasthenia gravis, and both PMBCL were strongly positive for mask at nuclear level, and 1 out of 2 endometrial clear cell carcinoma also showed a strong positivity for mask, the one in figure G, while normal endometrial tissue did not express mask protein. And so our data show nuclear translocation of the whole mask protein, or at least its extracellular region, and this mechanism is well known in the oncological field for other tyrosine kinase, and it is called memory receptor in the nucleus, and it is associated to cancer survival and also resistance to chemotherapy. Here are marked with a star all the tyrosine kinase that perform this mechanism, and probably we should add a star also to mask. But on the other hand, the detection of mask expression in cancer cells of patients with mask myasthenia gravis may support hyperanaplastic etiology. So the aim of this study is to investigate the role of mask protein in oncogenesis and its contribution to the development of mask myasthenia gravis. First, we will perform tumor tissue macroarrays to investigate mask expression across various cancer isotypes. Then we will assess mask expression in cancer-derived cell lines in order to perform ChIP-seq to clarify the role of mask in oncogenesis and mask interacting target genes in the nuclei. And lastly, we will perform conditioned medium experiments from cancer-derived cell line expressing mask to assess effects on IgG4 and mask antibody production on PBMC from mask MG patients and healthy subjects. And $10,000 will cover the purchase of tumor tissue macroarrays, cancer-derived cell line, ChIP-seq, commercial anti-mask antibody, and conditioned medium experiments. Thank you for your attention.
So we'll open up to the sharks for some interrogation. Who wants to go first?
Okay. Shall I start? So what you're going to look at is mostly a lot of associations between mask and things that you find in cancer cells, but how are you aiming to go from just associations to actual causal relationships?
The finding of mask expression in cancer cells of patients that develop mask myasthenia gravis after cancer diagnosis support hyperanaplastic etiology in some cases, of course not all the cases, and in a recent work we found that age at onset of mask myasthenia gravis was associated with cancer presence in a cohort of 94 patients. For example, the patient that was positive for mask that had endometrial clear cell carcinoma was 72 years old at mask MG onset. That is quite strange because mask MG onset is around 30 years old. So probably in some cases there could be an association. And another thing that we can do to demonstrate the causal association is to investigate the presence of B cells in the tumor. But of course, we need a tumor and a patient with mask myasthenia gravis, a fresh tumor. And to see if there are B cells in the tumor that produce mask antibody.
So I'm a shark, so I'm going to be a bit mean here and ask a follow-up question. So that's still all associations. So how do you know what is the chicken and what is the egg?
Yeah, but if we take, in the last experiment, we will take the surnatant of cancer cell expressing mask, and we put the surnatant with the PBNC of patients, of health subjects, to see if they promote a switch to Th2 response that is associated to the production of IgG4 antibodies. And even more, we also want to see if they stimulate the production of mask antibodies from PBNC of patients with mask myasthenia gravis. And for example, in PMBCL, there is a mutation of a receptor of interleukin 4 that promote the production of CCL17 that is associated to the production of IgG4 in patients with IgG4 disease. So probably there is an association because of the cancer microenvironment.
So I'll be the practical shark. What other resources are needed? I mean, the budget really is just for consumables. How will the rest of the staff, etc., be funded for the project?
Actually, we will cover the cost of all the material that I've shown, and if we need more funds, we also have funds from our institution, and I mean the project will be done at our university. Just the chipsec will be done in a different company because, of course, is a high-level expertise technique. But I have asked for a quotation, and they told me that it is around $5,000. Or if we need more money, we have our phone.
And it's a very ambitious project. If it all goes well, what does a good outcome look like? What do you have at the end of it, and what do you do next?
Hopefully, we will discover a new oncological pathway because probably mask also is involved in the cancer survival, like other tyrosine kinase, for example, EGFR or ERB that are target for oncological therapy. But on the other hand, we also need a cancer-tailored screening for some patients with mask myasthenia gravis. We don't know because it has never been explored before. And
so how does the result of the study help determine the screening you need to do then? I don't understand.
We can identify some subjects that are at higher risk to have the cancer, and so we can provide, for example, cancer screening like for other paraneoplastic disease, like autoimmune encephalitis or other paraneoplastic disorders.
So last question, are you more interested in cancer or myasthenia?
Myasthenia, but I think it's really interesting that mask protein that is involved in myasthenia could be involved also in cancer, so maybe there is a link.
So I'm sort of the dim shark here. So how common is cancer in musk. Right, there are a few case reports, but this common, because this is really based on, if I'm understanding, you've done this work on 2 patients.
Actually, MuSK is a very rare disease, and we were lucky because we explored cancer frequency in 94 patients, and we found 15 cancer in 13 patients. So the rate of cancer, of course, is low, but the expression of MuSK in cancer cell may suggest a paraneoplastic etiology in rare cases, of course. For example, PMBCL also is a very rare cancer, and the association to MuSK myasthenia gravis, that is another rare disease, is quite strange. So we were curious about that.
And in the cohort, the timing of the cancers relative to MuSK development, are these people who developed MuSK in their 20s and cancer in their 50s? I mean, how did you look at that?
These 2 cases both developed.
I'm sorry, I'm the interrupting shark. I meant in your overall cohort.
Okay, in the whole core we have 5 cases before 5 cancer diagnoses before MuSK-MG onset. 2 cases were concomitant, and in 8 cases cancer was after MuSK-MG onset. But of course, for a paraneoplastic disease, we just need one cell that expresses the antigen. So, for example, we don't know if in some patients the immune system was able to kill that cell, but it triggered the disease. We don't know.
Can I interrupt the interrupting shark?
Yes, but I might interrupt you.
We can help you maybe a little bit, because we have a lot of experts in the audience. So who of you is treating MuSK myasthenia patients? And who of you has seen cancer in these patients? Oof.
Who is what?
But where is who? So that she's a trial lawyer. Isn't that what that looked like to me? So I have one more question. So if I understand correctly, you're going to do your tissue microarrays on a subset of different cancers. How are you selecting them? For instance, are you looking at, and these are cancers presumably not from patients with MuSK. These are just from a tumor registry of some kind?
First, because we want to explore as cancer as possible, because we need to find a cancer line that expresses MuSK. So in commercially available cancer line are not so easy to find. We found one line of endometrial clear cell carcinoma in Japan, and another like PMBCL in Germany, but since it's not easy to order and to have a cancer line, we want to be sure to explore the expression of MuSK across various cancer isotypes.
Did you select based on your cohort of patients who had coincident cancer in MuSK?
Yeah, we just order. We just want to order microarrays that cover as much cancer as possible, and also the cancer that we found in our cohort of MuSK patients.
One last question. How many samples are you planning to analyze?
In the microarrays?
Yeah.
Microarrays, we will cover 80 cases of epithelial cancers, and other 20 cases of immune system cancers. And also we want to order a microarray for clear cell carcinoma. And then we just want to find one cell line that expresses MuSK to perform the conditioned medium experiments and with the PBMC of 5 MuSK-MG patients and 5 LT subjects.
Well done for the robust responses there, opening shots from the sharks fired. Do we have any questions from the audience? We got one here, Michael Hayher.
Hi, nice presentation. I'm just curious if others in the audience are actively screening your patients with MuSK or acetylcholine receptor antibody myasthenia for cancer, because I know that I'm not. and this would change my practice.
You can't find what you don't look for. I think that's a good point. Any other questions? Just one sec, Rick.
She's coming.
She's coming.
I guess the question I would have for you, based on Mike's question, is it was asked, what do you think could be an outcome of this study? Well, you did give a good answer to that, but the big picture, if you found a very close association, are you advocating then that that would lead the field to start doing cancer screening on every MuSK patient? Is that where you're going with this line of?
Yeah, it will be relevant, of course, for patients because if we need to do a cancer screening, we should know to do it and in which patients we have to perform it.
And then one last question, yeah.
A much easier way to look is, and which is pretty common, is look at your cancer population and see what percentage or how frequently you see MuSK antibodies in cancer patients. Have you looked at that, or are you aware of anybody? Because that's an easier way to look.
Yeah, we don't have to. We didn't look at it, but it is a good point.
Very good. Sylvia, thank you very much and well
you.
done. Great. So that's a fantastic start. The bar is set very high here. So we'll move on now. We're traveling across to Northern Ireland. Grace McMacken, exploring autoimmune myasthenia gravis in FSHD, a pilot study on a novel disease association. And Grace is joining us now.
Good morning, everybody, and thank you so much for the opportunity to present this idea. I'm Grace McMacken. I'm an early-stage clinical academic neurologist in Belfast, Northern Ireland. This is my first time at the NMSG. It's been a really amazing meeting with loads of fantastic presentations. I've also had really loads of really useful discussions with many of you over coffee and lunch that have helped lots of my ideas. And in fact, it was those types of discussions at UK myasthenia gravis meetings that led to the chance observation that many myasthenia gravis clinicians in the UK were caring for a patient who also had a diagnosis of FSHD. So we thought this was interesting, and we looked into it further using the UK myasthenia gravis database and confirmed 8 individual cases of FSHD with a co-diagnosis of myasthenia gravis. Even if we conservatively use upper bound prevalence estimates of 12 per 100,000 for FSHD and 20 per 100,000 for myasthenia, the expected number of co-occurring cases in the UK population of 60 million, if this was by chance, would be 1.6 cases. So our figure of 8 represents a 5-fold increase on the expected number. This is statistically significant and strongly suggestive of a non-random association. All of our cases had FSHD1 and then typically more than a decade later had gone on to develop acetylcholine receptor antibody-positive myasthenia gravis. The majority were male with a mean age of MG onset of 55 years. We feel that this is important and that it warrants further investigation. Firstly, this has direct clinical relevance. Of course, MG could easily be overlooked in a patient with FSHD due to the overlapping clinical features, and that's a missed opportunity for treatment. Perhaps even more interesting is the potential mechanistic relevance here for FSHD, but also for MG. Studying patients with FSHD who go on to develop MG gives us a unique window into the earliest triggers of autoimmunity, which remain incompletely understood. DUX4, the apparently expressed transcription factor in FSHD, has profound immunomodulatory effects. We know in FSHD muscle it drives MHC class I upregulation and muscle inflammation. It's also been shown to bind to the STAT1 promoter and thereby impair the induction of interferon-stimulated genes. Intriguingly, Whilst DUX4 is epigenetically repressed in most other adult somatic tissues, it is expressed in the adult human thymus. But its physiological role there and exact site of expression are not known. AIRE, deficiency of which is implicated in MG, also interacts with STAT1 in medullary thymic epithelial cells in one of several pathways regulating the elimination of autoreactive T cells. The fact that DUX4 can disrupt transcription at AIRE-regulated promoters, the very pathways known to be implicated in the pathogenesis of MG, raises the possibility of a convergent mechanism here. With your help, I would like to characterize this further. We do recognize the potential for ascertainment bias in the cluster of cases that we've identified so far from the MG specialist centres. Therefore, we'd like to start by confirming whether there is indeed a true increase in prevalence in MG in FSHD patients. To do this, we'll start by querying the UK FSHD registry, which holds comorbidity data on over 1,000 FSHD patients. We'd also like to start to try to explore the mechanism of MG in these patients. So we'll seek additional cases through the EuroNMD European Reference Network. And if this became an NMSG study, we'd also like to use that network to identify additional cases of dual-diagnosed FSHD and MG. We'll phenotype those cases, including review of muscle biopsy and imaging where available, and we'll also collect serum samples through the Newcastle John Muscular Dystrophy Research Centre Biobank. Samples will then undergo antibody profiling at Oxford Immunology Lab. We hope that through doing this, we can start to build a picture of the MG subtype that we're seeing in these FSHD patients. With your support of $10,000, we can cover registry data extraction, serum sample handling and shipment, and antibody profiling at Oxford. In summary, I've shown you preliminary data suggesting an increased prevalence of MG in FSHD. I'll deliver a 2-arm study to confirm this prevalence and explore the mechanism. Uncovering this link is important because it has direct patient benefit, but moreover, studying these patients offers the opportunity to explore the earliest triggers that initiate the acetylcholine receptor antibody response in MG. Thank you so much. I'm happy to hear your thoughts and questions.
Fantastic and perfect timing there. Excellent. To the second. So who's going to take this one on first? The sharks are circling. Go on, Jasper.
Thank you very much. Great. Again, some practical questions. So you're planning to test FSHD patients without known myasthenia for antibodies, or did I misunderstand?
No, sorry if that wasn't clear. So no, we don't think that would be particularly high yield at all, and it wouldn't be a good use of the money. So we're really just looking into the patients that have dual diagnosed cases already. We think that's much more interesting in to be able to pick out the mechanism. And we'll also screen unselected cases who have a clinical diagnosis of MG, and that's from the UK registry. So we're not screening any antibodies in FSHD patients.
So patients who have MG, you'll screen them for FSHD? Is that...
That's not part of the plan either. It might be something that we look at in follow-up studies, so whether there's a difference in D4Z4 transcript, for example, in MG patients would be really interesting if this did show interesting preliminary results. But no.
Right. So really looking at the patients who have both already, they already have ACH antibodies.
Yes.
But you want to do more extended antibody profiling, is that right?
We do, yes. So looking at for the other antibodies that we've suggested here gives us more of a sense of the potential etiology of the MG in that patient. So we see a different antibody signature in different subtypes of MG. And for example, if we saw fetal ACHR, that might point towards a more muscle regeneration, peripheral immunization as the etiology of the MG in these patients.
And so again, the same question as before, at the end of the year, if this is all successful, what implications would that have for practice, or what would be the next step, or what useful thing have we got from this apart from an interesting association?
So I think we might get a sense that there's a certain percentage or a certain clinical subtype of FSHD that is at higher risk of MG. I don't think we'll ever be warranting acetylcholine receptor antibody testing in all our FSHD patients, but we might have a different index of suspicion for a certain subtype of FSHD patient if this did yield interesting results. It also, we hope, would provide interesting pilot data that would allow us to go on to do further studies to explore the mechanism in more detail.
So I'm curious about the ascertainment bias, which was a question I had coming into this. And as I understand it, you're just going to try to get a larger data set to explore the association, right? So how is that addressing ascertainment bias? Because now you just have a much larger group of people who have the same bias. And did you think about using other large data sets, like a health services approach, to try and answer that question? And then a follow-up related to my fellow Sharks question. So you're proposing to do the additional immunologic testing on patients you've already identified, or are you planning on doing that in patients identified through, leveraging your friends network?
So coming to the first point, we're looking through patients who've been captured not through their myasthenia diagnosis, but through their FSHD diagnosis. So we have a different, those patients have, I suppose we have a different index of suspicion for diagnosing MG in those patients. So it is a slightly different approach. Ideally, something done on a population level, looking at GP databases or claims records or something like that would be amazing to do. And we would be really hopeful that we'd be able to do that in the future if we got interesting preliminary results. In general, those databases are all quite expensive to access. They wouldn't be covered in the cost of this pilot study, but it would be definitely something we'd love to do in the future. Remind me, sorry, of your second point again. So,
yeah, the testing, you're doing that just on the ones you've identified?
Yeah, so yes, it's only on cases of confirmed dual diagnosed patients, yeah. And again, it's just to build up a sense in this very pilot study, do we know what the potential etiology of the MG is in these patients?
So I wanted to ask you a completely different question. So those 8 cases that you identified, do you know their D4Z4 repeat sizes?
Yeah, I have all of that information. So all of them had less than 8 D4Z4 repeat length.
I'm asking because especially the longer repeat sizes, the 8 to 10 units, are found in about 1 to 2% of the general Western population. Yeah. So that might
absolutely. So
be an issue.
Yeah, absolutely. So all of our patients had a D4Z4 repeat length clearly in the pathogenic range. So there were no gray zone patients. They all had typical FSHD for many years. So I don't think this is false positives or spurious results. These patients have all been examined in specialist centres with a clear diagnosis. The other thing, just to come back to that, I suppose, is that is kind of interesting, maybe, and a follow-on study is we do have a high proportion of patients in the general population with this grey zone D4Z4 repeat length, and it would be really interesting to know if they're at higher risk of developing MG.
And then, again, let's use the fact that we have a whole community here. So, again, who in the audience has patients with both FSHD and myasthenia gravis? You might have something here.
I'd be very interested to speak afterwards.
Did you check who was raising their hands? And then one final question. So if the clinical takeaway is don't miss MG and FSHD, then does it really require an entire research project, or is just a simple message stay aware?
So I think that's one of the clinical takeaways, and I think having a defined increased association, even if that is even if it's 0.5 or 1% of FSHD patients, that's still really important. I know in my country, a lot of FSHD patients are not managed by neuromuscular clinicians, they're managed in general neurology clinics. So having that different index of suspicion, looking specifically for chewing fatigue or extraocular muscle involvement would change our management, I think. But conversely, what's potentially even more important with this work is the relevance for the pathogenesis of MG. So whilst, yes, there is that direct clinical relevance, maybe from this pilot study, a follow-on study would really go to that and could have even more relevance there, I think.
Hi, Grace. I wanted to let you know that prior to coming here, opened the UK FSHD website that they have already publicly available, all the questionnaires that they've been asking. And I haven't seen any question relating to myasthenia gravis in this population. There's only one small open, one small box that they can mention any other conditions. And I don't know how many characters it also involves, and if the patients actively know that they can actually input this kind of condition in this little box in one of the questions of many questions of this registry. So how confident are you that it's these conditions being captured in this registry? And maybe you should seek more relative databases, like it was mentioned before, like the UK CPRD that collects data from from all the health records around the UK, which could actually tell you more information about this population.
Yeah, absolutely. So there's been a new iteration of the UK FSHD registry that's only just being published. So I spoke to them last October about this idea and made sure we got myasthenia gravis listed as one of the drop-downs in that new iteration, as well as other autoimmune conditions. So patients will have the tick box to be able to definitely write in their myasthenia gravis. They'll also be able to write in some free text about that. And then what we would like to do is distribute a survey to the patients that click the myasthenia gravis button to be able to get some more information and consent them to de-anonymization. That will also allow us to do some capture-recapture on the patients that we've already identified to make sure we're not overestimating our numbers. And yes, absolutely, it would be fantastic to look at bigger databases. They are expensive because we've looked into that, but it would be brilliant to do in a follow-on study.
So, I mean, one, I think you just need a friend who has the access, right? So I think finding the right collaborator might be helpful there, and I'm sure there are people in the room that might help. I guess the other question is, could you use the Clinical Trial Research Network? I mean, you're going at a patient-facing way of doing it, which is great, but, I don't know how many people are participants are in that network, but presumably quite a few. They're very well phenotyped and seen by experts in this room. That might be another resource you could use to look at it.
That's a really great idea. I know just anecdotally of a few cases that have been excluded from natural history studies and clinical trials because of their dual diagnosis. So that's just maybe a slight caveat to that, but yeah.
Yeah, but I would imagine they might have these sort of screen failed data for you for that reason. I just don't know.
Fantastic. Yeah, that's a great idea.
Jeff, I suppose.
Yeah, thank you.
Okay. Do we have any questions from the audience? We've got...
Dr. McCacken, you are onto something. This is a real thing. Years ago, Valeria and I think it was, I don't know if it was this meeting or at some meeting where it was probably here, we both were sharing with each other that we had cases like this, and we wrote them up. I can't remember where we wrote the cases up. 2 cases, yeah. But, and then in Columbia, Missouri, I have a patient with both. And he told us that he has this autosomal dominant FSHD family history, and 2 other family members have MG, proven. So there definitely is an association. And we wrote this up in the RNMF Neuromuscular Journal about a year ago. I had one of our students write it up, and we hypothesized why this might exist. You could probably dig into that and lend some light into a possible pathogenic course for why these things exist. I guess the question is, let's say we go down this pathway. You find 20 what what do you think that will do for our field of neuromuscular disease? How will it improve where we are at today if we say, okay, instead of 8 cases, we know now there are 50 cases worldwide? What's the implication of that?
Yeah, so I think to date, essentially, we have lots of case reports, and yours have contributed to the literature as well on these coexisting cases, but we've never really looked at a population level. We've never done epidemiology, although it is challenging in these ultra-rare conditions, to show that there is a true increase in prevalence. So that's kind of what we're attempting to do. It's not perfect because it's challenging with the rarity of it. But really just to give more of a sense of, is there a true increase in prevalence? Is this just lots of sporadic case reports because people are looking for them, or really is there a true disease association? And I think that would change our management. I think we would think about FSHD patients, maybe male FSHD patients in their 50s. We might think about them slightly differently if the disease trajectory isn't what we expect. So I do think there would be clinical relevance there. But the other thing that's maybe a little less direct is DUX4 implicated in the pathogenesis of MG? And does that give us more insights into the earliest triggers that lead to MG?
And Valeria, did you want to follow up on that?
Yeah,
just quick fire if we can, and then one from the back.
No doubt, I'm having my shark glasses, even though I'm not a shark. No doubt that the association is interesting to understand the mechanisms. I am not sure that this type of investigation into the associations cannot just rely on looking first at the networks and just finding all the patients there. And I think this would be a second step of investigation to try and investigate. First, I'd like to see a greater number of cases there and stratifying by the patient's features, because FSHD mechanisms are very complex. And so I'd like first to see a preliminary work with the existing networks there.
I have a comment suggestion back here, if I can just add that to the
Yep.
area. So I think it will take you 5 minutes to use a greater database like TriNetX, for example, and just to look at prevalence. And then I personally don't have a patient with both diseases, but I have other neuromuscular disease patients that have myasthenia gravis. And I think I just check broadly, because whenever there's fatigue, and obviously FSHD has a lot of fatigue, I check for myasthenia antibodies. And so there is ascertainment bias, but I would encourage you maybe to use another disease control as well, because other muscle diseases also have fatigue and maybe are more likely to be checked for myasthenia. So you might want to look at not just FSHD in these databases, but other muscle diseases like myotonic dystrophy. I also invite you to use the national registry from the US. We have hundreds and hundreds of FSHD patients, and it would be very easy and quick to do a search there as well.
That's fantastic. Thank you so much. And yes, we agree with the idea, absolutely, of a disease control. I think particularly if we were able to do the antibody profiling and it pointed towards this being a peripheral immunization due to the inflammation of muscle, it would be really nice to look into other chronic muscle diseases that have a component of inflammation to see if there's a tendency towards acetylcholine receptor antibody production in those patients as well.
Brilliant. We can see a collaboration happening before our eyes here. That's fantastic. Good. Thank you very much, Grace.
Thank you.
We'll move straight on to our next talk. We're going back to Italy now. We have Laura Tufano, GI involvement in myotonic dystrophy type 1, characterization of a variable phenotype by PROMIS GI. So Laura, over to you. Thank you very much.
Hi everyone, and hi Sharks, and thank you for the opportunity of being here today. Sorry. Okay. So myotonic dystrophy type 1 is the most common muscle dystrophy in the adults. And beside skeletal muscle involvement, it involves also smooth muscle, causing, among others, gastrointestinal symptoms. And here you can see the most common gastrointestinal symptoms in DM1 and the relative prevalence estimated in literature. And as you can see, the prevalence is quite wide, and this is because we have many limitations in the assessment of gastrointestinal involvement in DM1. And the most important is the fact that we are missing a standardized and validated tools to assess gastrointestinal symptoms. And this creates a huge unmet need. On one side, the phenotyping of gastrointestinal involvement in DM1 is poorly characterized so far. And on the other side, even most importantly, we are currently performing phase 3 clinical trials with DM1-specific therapies with the potential to target smooth muscle, but we are missing a tool to evaluate gastrointestinal symptoms and to assess the effectiveness of these therapies on the GI tract. So this is why the aim of my study is to assess the prevalence and severity of GI symptoms in DM1 with the use of PROMIS GI scales. What are the PROMIS GI scales? So there are 8 patient-reported outcomes. They were developed about 10 years ago, in English, of course, and 4 of them have already been translated into Italian. And I chose the PROMIS GI scales after a literature review of gastrointestinal involvement in DM1, looking at what have been used so far, and also of GI questionnaires in general. And in the end, I ended up choosing the PROMIS GI questionnaires because these are the pros of these PROs, in my opinion. So they are very comprehensive because they cover most of the most common GI symptoms in DM1. They are adaptive, meaning that they are built with an item skipping logic, so that not every patient will answer to every item to all the questionnaire, but they answer only to the items that apply to them. And they are standardized and they are responsive to change. My project would involve 2 neuromuscular clinics in a cross-sectional study. One, the University of Rochester, New York, where I recently spent 8 months working with Dr. Hamel until last May. And the other is the Sapienza University of Rome, where I'm currently performing my PhD with Dr. Matteo Garibaldi. And I would collect also other characteristics like demographic, genetic, DM1 clinical features, a feedback questionnaire on PROMIS GI, and also a structured GI interview to cover GI manifestations that are not included in the PROMIS questionnaires and medications in use. So these are the project steps and the timeline in months. This is the flow of the events. So you can see that most of the time at the very beginning is dedicated to the translation of the 4 PROMIS GI questionnaires that need to be translated into Italian to be administered to Italian participants. And then as soon as this is done, the data collection in Rome can start. At the very end, we will have the data analysis. This is an overview of the cost. As you can see, much of the budget is used for the translation of the questionnaire, which is a long process, which needs to be followed by Health Measure, which is the center providing the PROMIS GI questionnaires. And as you can see, the data collection in Rochester is for free, now because we already started it when I was there. So now I'm going to show you this preliminary data on 24 DM1 patients who filled in the PROMIS GI questionnaires, and 62.5% of them reported at least 1 GI symptoms, and here you can see the prevalences of the GI symptoms and the severity into mild, moderate, and no severe case. So at the very end, why should you fund me? And there is an orca because orca are primary predators of white sharks. So there is a huge gap, and I hope, which is very important to patients, and it is totally understudied so far. And the need is very urgent. Actually, we needed it yesterday before the start of phase 3 clinical trial, so we are already missing out. And the strategy is successful, so it's fast and feasible, as my preliminary data showed you. And last but not least, collaboration first, so this project may foster transatlantic relations. So thanks to my teams, both in Rochester and in Rome, for their work and support. Thank you for your attention, and hope that you would like to launch my idea with some true seed funding.
You want to go first? No? I was trying to convince Nicolette to go first.
I wanted to ask, are you going to include all onsets of myotonic dystrophy in your study?
No, this is focused only on myotonic dystrophy type 1.
Yeah, but
Okay.
all the onsets of the disease, or congenital, juvenile.
Yes, I would like to collect data from all of them. I am mostly from non-congenital DM1, but I would focus mostly on non-congenital DM1, but why not also collecting data? But when I wrote 150 patients, 75 and 75, I was thinking mostly of non-congenital DM1.
Okay, I also wanted to ask, are you planning to use any quality of life questionnaires like MDHI in order to understand if these symptoms are also affecting their lives in which extent as well?
Okay, so the Promise GI questionnaires, there are 8, they have different length, and actually among the questions that they include, they ask not only how often a certain symptom occurs, but how much the symptom is bothering the patient, and how much is the impact of the symptom on the life of the patient. I'm not planning to, I didn't look for other questionnaires, but this could be a good idea to add also another one. Yes.
Okay, so the last question I wanted to ask. So the Promise GI, it's basically it's questionnaires that they were initially done on adult US population,
Yes.
and it asks questions about symptoms in the past week.
Yes.
Some questions, for example, I could only access the swallowing questionnaire from EU, so I don't know if it's accessible. All the questionnaire, all the questionnaires are also available. Some questions like how were your symptoms, if you had any vomiting or any other issues in the past 7 days. It's not quite applicable to our population because some of them have pegs, and it doesn't have any tick box to mention that it's not applicable for this population. How are you planning to address this?
Yes, yes, thank you. This is totally true. Among the limitations that I was thinking ahead, I could see that Promise GI questionnaire always asks about symptoms in the last 7 days. So this is why we decided to add also a feedback questionnaire on Promise GI. Maybe if the questions are about constipation, the patient may report that they didn't have constipation in the last 7 days, but in the previous month, for example. And also the other thing that is a possible limitation is the fact that I want to collect medication in use. For example, if you notice the prevalence of reflux is 1% in 24 patients, is only 1 patient, but of course this is because maybe they are taking medication that may cover the symptom. So yes, this is something that we want to deal with an interview, structured interview, that like the other data that we want to collect, like a structured interview focus on GI manifestation in general. So not only trusting to add some data other than the Promise GI questionnaire, if they were missed.
Okay, then let me ask the obvious question that we always ask when talking about DM1. So this is a difficult population because they underreport everything. So are you sure the patients could tell you that their gut is fine while they're eating soup through a straw? So how are you going to account for that the patient just won't tick all the boxes, I'm fine, I'm fine, I'm fine, because that's what they do.
Yeah, this is true. Sometimes DM1 patients may underestimate their symptomatology. And so this is why we also add the interview and try to understand if there is something that was missed that was not evident in the questionnaires. But at the same time, we know that they are aware of their GI symptoms that are very prevalent. For example, in the PRISM study, more than 80% of DM1 patients report that GI symptoms are very important. And sometimes I have experience in clinic also that patients also with a minimal muscle involvement, they report, for example, fecal incontinence that's so bothering that they cannot go out from home. And so I'm sure that...
So have you thought of any way to account for that? So have you thought of things that will help you also more objectively capture something on GI symptoms?
I still, of course, in the interview, it's different because I'm going to ask questions, and then I can also compare what I collected with what they, how they filled in the questionnaires. And other objective data in this, other than the interview, I haven't actually, it would be much a different project if I want to add, for example, I don't know, a quantitative study for reflux or of, I don't know, manometry of the rectum or something that is not included in this study, of course. But it's something that it would be useful to know if these measures work, because they could be added, for example, in natural history studies or in clinical trials that they collect PROs also to assess, for example, responsiveness on other outcomes that now the objective measures maybe are the muscle and function.
Yes, so one final question. So I've sharpened my teeth, but it may be helpful for you to sharpen your objective a little bit. Because in your presentation, your objective was to look at the prevalence and the severity. But then again, I hear you talk about clinical trials and natural history studies and validity and responsiveness. Those are two different things. So what do you want to achieve? Do you want to use this tool for getting a sense of disease burden? Do you want to use it as a clinical outcome measure? What is your actual objective?
To have an outcome measure that can capture GI symptoms in this population in a way that can be used to assess the both prevalence and severity of the symptoms, because if you look at in literature now, it is prevalences are very wide and the studies are very heterogeneous, and so I would like to have some clear data about how much the GI symptoms are present in DM1, and if this outcome measure works, I would like that to be also added in natural history studies or clinical trials.
Yeah, so maybe one thing to take from this session is those are two different things that need different measurement tools. So you may want to consider what you want to get out of it.
Okay, thank you.
Yeah, I guess it's a similar kind of question. You know, as you say from the start, there's lots of studies already that have looked at this and shown quite different prevalences of these symptoms, and your study will add another prevalence to the symptoms. And so I guess the output that these symptoms are common in DM1, we already know. So I guess, again, the question is, what's the next step? You know, once you've done this, what would you do next in this population and this question?
Something also maybe different from the previous studies in literature. I don't think this is only going to add another prevalence in the bucket, but because many of the studies I looked for to understand the prevalence of a certain symptom, for example, they had a certain population and they investigated for constipation, or they investigated for reflux or for lower GI symptoms. So in this way, I think that these questionnaires capture the symptoms from the oral cavity to down below, and so from the same population, we have a prevalence of all the most common symptoms of the GI tract. You know, sometimes in other studies, I found out that they report in that population only the prevalence of diarrhea, for example, without speaking about constipation. You know, instead, I would use the same measure to assess all the symptoms of the GI tract in the same population from the beginning to the end. And then, if this measure works and if it is going to give reasonable results, I would like that to be included in natural history studies or...
And just so I understand how it works, I was interested that 4 of them scales have already been translated to Italian.
Yeah, in
is
the
there?
past.
I mean, is it a proprietary scale? You have to pay to use it. I mean,
sorry, is there?
Is it do you have to pay to use the scale, or?
I don't have to pay to use the scale. I have to pay to translate the scale, which is not cheap.
It's quite expensive, isn't it?
Yes.
$5,000 to translate some questions?
I think it's even more, actually. But it is quite expensive, yes, because the health measure, which is the center providing the Promise GI questionnaire, they need to overview the process. It is also a long process, as you saw, 4 months. It is about like 12 weeks long, because they, even if the questionnaire may seem very easy, they want this process to be very precise, and so that then my translation, of course, will be available. They will use it also. If any other gastroenterology wants to use it for their symptom in Italy, they will be able to use. So it's not...
And one final question. Do you know if the prevalence in the healthy population of GI symptoms is different between Americans and Italians?
This is a very good question, thank you. I don't know. I don't know why I imagine yes, but I don't know.
Well, we have some Italians and Americans in the audience. Do Italians have GI? No.
Yeah, we've not had a show of hands, Carly. Do you want to... Gordon, do you have any quick questions?
I mean, I have the same thoughts of my colleagues. I don't work in myotonic dystrophy yet. I'm thinking about Chad Heatwell's really wonderful talk yesterday, and there's a GI subscore to the health index, and trying to understand specifically what you're trying to achieve relative to that, and whether, you know, this broader kind of survey approach is the right way of doing it, or whether a smaller focused group to try and address the problems that I think, you know, my colleague talked about, right? So I don't have more other than maybe thinking about more focused work ahead of time, and Chad's point that having something that's very tailored to this population would be very helpful, and you're using something that sounds like a great instrument but isn't tailored, so maybe tailoring it with a smaller group of people to address the current gaps that you see, and then scaling up would be something to consider.
I know about the MD high section taking care of gastrointestinal symptoms, but I wanted something more detailed to characterize more the symptoms, because yes, other than the 5-6 questions of the MD high, and to have more questions about a single symptom, maybe also to try to overcome the underestimation of symptoms by DM1. I don't know. Yeah.
A couple of questions from the audience, perhaps?
Yeah, I was going back to the section in the MD high on this part, and the fact that it's the promise is more general does not necessarily help to understand the details of the problems, because if you get even with a short version, if you see that's the problem, one of the issues in the population, then you will need more detailed, specific things on that specific area, like incontinence, like reflux, to get and some objective measures. Because otherwise it's adding details to what's in the network in 700 patients that can be looked at.
Okay, any other questions from the audience? Should we wrap up there? So thank you very much, Laura. That was great.
Thank you.
So last but not least, we have Carola Ferrari-Agradi from the Nemo Centre Milan, looking at cerebellar function in myotonic dystrophy. So Carola, over to you.
So, hi everyone, and thank you, Sharks, for the opportunity to present my proposal today. I'm Carola, and I'm a resident in neurology at the Nemo Center. So, CNS has been extensively investigated in myotonic dystrophy type 1, but pathogenetic mechanisms are still only partially understood in this disease, and consensus about how to assess CNS symptoms is still in discussion, we can say. So we all know that the cerebellum has an important role in movement coordination and vestibular processing, and these aspects have been investigated also through a DM1 mouse model that confirmed RNA toxicity in the glial cells associated with cerebellar dysfunction and motor incoordination. What may be less known is that the cerebellum has a very important role, not whole, as I wrote in the slide, in cognition and emotion. And as you can see, mainly the posterior lobe of the cerebellum are involved in controlling executive function, language and visuospatial processing, emotional regulation, and affect. Does these symptoms look familiar to you that follow people with DM1? So yes, there is some clinical overlap between the impairment in this area of the cerebellum and what we see in patients with myotonic dystrophy type 1. And these symptoms have a name, which is the cerebellar cognitive affective syndrome, which can be evaluated through a scale that my colleagues are distributing so that if you're not familiar with the scale, you can follow. And I have the scale.
And it's plastified. Is that a word in English?
Laminated.
Laminated, thank you.
So as you can see, this is a 10-item scale that can be administered in 10-15 minutes and provides you with a total raw score, cutoff diagnosis, and pass-fail criteria for each single item, providing a possible, probable, or definite diagnosis of CCAS. And as you can see, the domains explorers are among the most involved also in myotonic dystrophy. For example, you have cognitive flexibility, verbal and working memory, or attentional and emotional control. So my objective is to systematically administer this scale together with a validated battery of cognitive tests at the NEMO site and to correlate the score with clinical, neuropsychological, and neuroradiological variables. The participants will be 50 patients with adult and pediatric onset DM1, and they will undergo clinical assessments including neurological examination, assessment and rating of ataxia, PROs, including assessment of anxiety, depression, quality of life, and disease burden, a cognitive battery of validated tests, and of course the CCAS scale. And we will also do a 3T brain MRI and hopefully diffusion tensor imaging. So the funding will be distributed as follows. So for the protocol and electronic CRFs development, data entry and data management, and finally data analysis. So why should the shark's money follow this current? We will dive deeper into the cerebellar role in myotonic dystrophy, hopefully providing a new potential explanation for the cognitive abnormalities that we see in these patients and providing a potential substrate for neurorehabilitation. And of course, we will also have a new outcome measures that has proven to be reliable and reproducible, fast and bedside friendly, and what is more important, provides a diagnostic cutoff and a raw score to monitor over time, and maybe also before and after administration of disease-modifying drugs, which is very important in the era of drugs targeting also the CNS and crossing the blood-brain barrier. So I have just the last recommendations for you sharks, which is in the last slides. And thank you for your attention.
So I'll start with the budget question. So you're going to be doing, if I read correctly, 25 adult, 25 juvenile. Are you doing MRs on each of them? And I didn't see that in your budget.
Yes, because we have started a collaboration with the hospital and NEMO is inside. And usually in Italy, you can do just a basal MRI as a clinical practice investigation. So we have some data collected in the past year from DN1 patients that have been followed at NEMO for more than 5-10 years. Some of them already are undergoing brain MRIs, so we have some preliminary data just by seeing the images. And so it's something that we can do without putting that in the budget.
Well, it's good to be in Italy.
Just to 100% clarify, in the abstract you said you would do follow-up in 12 months. But
Yes,
that's presumably
so
not
the
planned for this.
beginning we will do just one assessment and then maybe follow up after 1 year, but it's just to see if the scale is valid and it's something that can see differences and it's like feasible and everything, so it's like a pilot. So for sure we will do one assessment and then of course we'll go on assessing.
I guess that's my biggest concern, is it's quite a ambitious study to with this funding, do 50 patients, all these tests, all the analysis in 1 year. So I guess my question is, what do you see as the biggest challenge to completing this project as planned, and how would you kind of overcome that?
So time, for sure, can be a limit, but we are very lucky at NEMO because we have a fantastic neuropsychological team. So we have 5 psychologists that are assessing CNS function in myotonic dystrophy, both in the outpatient clinic and in the inpatient clinic. So while the patients are in the ward, it's easier to have a broader battery of tests. But when we see them in the outpatient clinic, we have also the psychologist coming and doing some evaluations. So for example, we routinely administer the TMT A and B, the Stroop test, the Ray figure, and also the verbal learning. So there is many tests that we can do. And I was also in charge of doing part of the evaluations. So, and we also have many neurological residents. So I think it's feasible.
And again, at the end, are you suggesting that we use this a scale or score as a screening tool? It's quite a long test, or what do you think we'll do after this project?
No, actually it's quite quick to administer. So I was administering the test to patients just to have a general idea of how long does it take. And it's like 10-15 minutes because it's quite easy to administer. And so I think that it's very useful to monitor cognitive function, maybe also over time, because you have the cutoffs to put a diagnosis and have a general idea of the cognitive status of the patient, but you also have the raw scores up to 120, so you can also monitor that the patients maybe is pathologic or non-pathologic, but you can also monitor if their scores improve or not.
And I guess my final question is, if I understand correctly, this scale was designed for people who had a cerebellar problem to pull out their cognitive problems, but in DM1, the pathology is much more widespread in the brain. So would the more detailed psychological testing you're doing anyway not be more appropriate or more useful?
Yes, so what is interesting is that it was validated for cerebral pathology, but the patient involved in the validation study were not only like people with the cerebellar stroke, which is quite localized, but also with spinocerebellar ataxias that have a broader pathology. So that was something that I was checking at the very beginning because that was my main concern as well. But what I think can be useful is that, of course, with the data that I'm collecting at this very moment, I will never know if the pathology is coming from the cerebellum or the connections between the cerebellum and the brain. But I think that this is the first step. I plan to do some functional MRIs in the future so that we can target in a best way the pathology of the entire brain, but I think that in terms of monitoring of the disease, it can be useful anyway because the symptoms, the CNS symptoms that it targets are the ones that we experience in our everyday life with the patients. So I guess for the second part, it's going to be very useful as well.
A quick follow-up, because I was really curious. I'm looking at these and trying to determine whether I would answer them correctly, and I think I would do reasonably well. But has this been validated for cerebellar-mediated cognitive dysfunction in patients who have more generalized cognitive dysfunction? I understand it's validated in SCA, for instance, but in a group of aging adults or patients who might have coincident dementia, for instance. In other words, someone who has dementia is going to struggle mightily with this, but that doesn't imply they have cerebellar disorder. And obviously, this is a much broader cognitive problem for our patients than just
No, it's
cerebellum.
still quite new and not so known scale. For example, I was speaking to people in another hospital dealing with cerebellar ataxias, and they were not like administering the scale, so I think that it needs to be like pushed more. So I don't have data about other types of dementia and cognitive decline.
So 2 questions. The first one is, we all know that DM1 patients have cognitive issues, but whatever test we use, not specifically for cerebellar, but just for cognitive dysfunction in general, whatever test we use, it's so hard to capture it in a single test. So what makes you confident that this test will be able to capture it?
So it's one scale, but it's 10 tests in one. So that's, I mean, we are like looking at different domains in one shot. And I think that that was one of the most interesting things that I thought when I just jumped into this scale, because it's very quick, but you can look at different domains. The reason why we are also administering the already validated test is that we want to see if a test that is so brief and fast can give a general idea and so correlates with the test that we are already using. So.
And then one final question to take down your entire project. Why is this even important? Because once we have those disease-modifying therapies that will get to the CNS, we'll just treat these patients no matter if it's in the frontal lobe or in the cerebellum. Why do we care?
Yes, but in science you need to measure. So yes, so this is, and actually I think that it's important not only for the therapies that can cross the blood-brain barrier and target the CNS, but it's very important also for the drugs that maybe are not targeting CNS but are modifying maybe the neuromuscular symptoms, because, I mean, maybe this is too far, but what's the point of walking 10 miles instead of 10 meters if you have apathy and you have, you know, many cognitive symptoms that keep you in your house because you don't want to go out and everything. So I think that we really need to stress the investigation in CNS and providing outcomes measures also in that.
I do agree that CNS problems are a main issue for the patient, but why do we need to separate the cerebellum from the frontal lobe or other?
Well, that's true. Better. So the cerebellum has a potential for neurorehabilitation and also can be targeted through like techniques like the transcranial magnetic TMS stimulation. So it has a higher potential than the cortex or, you know, the subcortical white matter to act on. So that's something that is really interesting. It can provide like a new substrate for new therapies, and I think that's amazing.
Nicoletta, you go
in. You mentioned that you're also going to include children with myotonic dystrophy in this study.
So not children, but it's something that we are definitely trying to do next, because the scale is validated only in the adult population. What I would like to do is to, of course, include patients with a pediatric and an adult onset, and also some patients with a congenital onset that have more than 18 years old, because you know better than me that the pathogenetic mechanisms are a little bit different, and we still have to understand deeper what's going on there. So the idea is to have the broader population in terms of age of onset. So yes.
From my experience, when I had similar studies with congenital population, adult congenital population, myotonic dystrophy, a lot of patients that have we struggled to get them into MRIs due to pacemakers, due to them not being having claustrophobia and severe anxiety in being in enclosed spaces. Do you think that this will impose a risk in enrolling this population? Have you thought of any other alternatives? Because having them sedated is not the best way to go. I mean, take into consideration all this anesthetic risks in this population as well.
Yes, that's something that the problem we are struggling as a community. So we have started administering some basal MRI, standard MRI, to congenital patients that are adult now. And we didn't have that much problems in administering the MRI, while with children, that's a mess, a nightmare. So I have no other ideas to have like images of the brain, so I guess that we'll just limit to administering the scale and correlating it with the neuropsychological test, which is a first step.
Thank you.
Thanks.
And do we have any questions from the audience? We got one right at the back. And if anyone else has one final question after this, get your hand up.
So, thank you for the interesting talk and proposal. I have two comments as a former ataxiologist about the scale. So the first one is that we tested the scale in the Italian population, the CCAS, and we found it's unpublished data, at least it was until I was working in ataxia, but we found that there is a high number of patients that were false positive in the control group. So maybe you want to consider also that. And the other thing is regarding the SARA scale, because of course you want to be sure that there is maybe some kind of cerebellar impairment and to test it with the objective measure motor scale. So SARA scale is also very sensitive, so it's not difficult to get a 3-point for SARA if you have some kind of muscular impairment, like for the heel shin slide, tremor, also with the walk. So this is something that must be carefully accounted for.
Yes, thank you for your comments. Actually, while administering the scale to the DM1 patient as a preliminary investigation, I also saw that the rating of positive scores was quite high. My idea is to ignore people that score 1 or 2 out of 10, but taking the ones that have at least a 3 out of 10, so to be sure that there's something there. And of course, the correlation with the other neuropsychological tests is something that we'll take for sure into account, just to be sure that we are doing right. Coming to the SARA, I'm glad to take any suggestions from people that do ataxiology, because I'm not an expert in that field. So I know that like tremor and muscular impairment can affect the scale, so I'm looking at possible strategies to minimize that effect. So if you have any suggestion, we can have a talk later and discuss about it. Thank you.
We have used SARA in myotonic dystrophy population, and we have also published a paper on it, so you can also take a look at it.
Thank you.
Very good. Another collaboration before our eyes. Yeah, great. Any last questions? I think we'll wrap up there if that's okay. So thank you, Carola.
Thanks.
So that was 4 fantastic presentations, and I think we should have another round of applause for the presenters. I think it takes a lot of effort to do that. I think they did really well, and to stand up here and respond to questions in such a sort of efficient and yet polite way, I think, and robust way. I think I'm really in awe of the young investigators. So well done. So the Sharks will do their scores. We'll top them all up, and then we'll get back to you after lunch. And I think you got 15 not lunch, just coffee, I'm afraid. You got 15 minutes for coffee. Thank you very much.
If we could have everyone return to their seats so we can commence the next session, that would be very much appreciated. We'd like to stay on schedule, and we have a very exciting last session. Thank you.
Last person talking was taller than me by quite a bit, I think.
All right, so thank you everybody for coming back and lasting till the end of the meeting. We have a really cool session at the end here where we're going to talk about some caution and interpretation of clinical trials. We have Dr. Katz from Sutter Health who's going to be talking about issues with trials and efgartigimod in CIDP.
Well, thank you. I'm going to apologize for, in case I go a couple of minutes over in this talk, because 20 minutes is not a lot of time to do this discussion, which probably took me about 100 hours of thinking. But this is about a trial design in CIDP. And before I want to start talking for 2 seconds about trial design, I just want to talk from a clinician standpoint of what it's like taking care of CIDP patients. CIDP is a nerve disease, for those of you who don't know. Do I have a clicker to change the?
Should be on the.
I think the last speaker took it. Yeah. I could do my other Italy speech, friends, Romans, and countrymen. But I don't have a...
Yeah, we're going to look for that, but in the meantime, you can just say next slide, please.
I think it's going to be hard the way this talk works to do it that way, to be honest with you. There's a lot of...
well, could
so I'm just
Ms. Ferrari
make
or
everybody wait.
Whoever has the clicker, bring it back.
How did it switch, though, if there's no... Oh, you did it. Oh, here it is. Yeah. All right. Fell into the crack. Yeah. All right, very good. My wife is in the audience thinking, what an idiot. Anyway, so before I start talking too much about trial design, the clinical hat says that when you're taking care of this, there's two populations of patients that get referred to with CIDP. One population has the disease, and these are chronic patients. You take them off their drug, they relapse. The other population, CIDP has a big problem. Neuropathy is a very hard field diagnostically. There's a lot of patients running around with CIDP who get referred to. We have a very high-level tertiary clinic, and they either don't have the disease or they're in remission. And in those two categories, it is very hard to get these people off their drugs despite they've developed a relationship with their drugs. So if you imagine if these patients go into clinical trials and have this type of relationship with their drugs, it's going to be hard to get them off their drugs, and they're going to react to the trial in ways that might not be so obvious. So with that in mind, I'm going to go through what I think was sort of an imperfect trial design. These are my disclosures, and I left one off, but it doesn't matter. But the disclosures are important slightly, which is rare for people to say, because I've been very involved with being an adjudicator in CIDP trials. And adjudicators are people who get to decide when somebody gets submitted to be part of the trial, you get to decide if they really have the disease or not by reading their records and working with other adjudicators who happen to be big experts in the field. And the concept is that these experts are going to solve the problem, but maybe they don't. So let's run through this. Here's the results of this trial on efgartigimod. Efgartigimod is an FcRn inhibitor, so it lowers immunoglobulin levels, and that's supposed to help the disease. The trial took patients, and I'm going to tell you all about this as we go forward, who are already responding to the research investigational product, and they stopped it, and the top line is the placebo group. The top line is the treatment group, and they didn't relapse over 48 weeks. And the bottom line is the placebo group, and they did relapse over 42 weeks. And that had a p-value of 0.0001, and the FDA approved the drug, and everybody lived happily ever after until I came along. So this is the withdrawal log. And I became curious because we had, number one, I had all this background in the disease, but number two, we had enrolled 4 patients in these trials at our center, not just this trial, but another one with the same category of drug. And our patients had these very poor outcomes when they're treated. And it wasn't that they didn't respond to therapy. It was that they ended up in hospitals. They ended up unable to move, paralyzed. So we got worried, stopped enrolling patients in these trials, and sort of sat around like, ha, that was fun. And then the result of the trial came out about a year later, and I was a little bit shocked that the drug could work that well. So I did some math, and my curiosity got piqued, and I ended up writing the editorial I showed you in the last slide. But This is from the company on the right, and the purported response rate to this drug in a Boolean they respond or they don't respond sort of way is 67%. So 2/3 of patients respond, which means 1/3 of patients don't respond. I had seen not just non-responders, but these poor outcomes 4 times. 1/3 to the 4th power is 1 out of 81. So with a 1 out of 81 chance, was I fooled and tricked? Or does the drug not work? A 1 out of 81 chance can happen to anybody, but it's never happened to me. I've never won a lottery, so I became skeptical. So I'm going to tell you about this trial design, and we're going to go through it really fast from left to right, and then we're going to sort of delve into what might have gone wrong. So what you do in a trial like this is you start off with some tasks to figure out who has the disease and who doesn't, which is important in CIDP. And those tasks, without thinking about it, are selecting sites, because you want the sites to know what they're doing, choosing criteria that define the disease, and then you send it to your experts, your adjudicators, and they figure out who has the disease and who doesn't. The next thing you want to do is figure out if the disease is active, because there's patients out there who had autoimmune activity and now they don't, and they're on the drug for no reason for years. So you don't want inactive patients in your trial. So you have a run-in where you're going to stop the drug. Then you're going to rescue everybody, because you don't want people off drug for a really long time before you randomize them. So you rescue everybody with the drug. You take the 67% of patients I just showed you and responded, withdraw half of them, put the other half on placebo, and then the winners get an open label extension and get to live on the drug happily ever after. Brief comment on the open label extension. A lot of these patients are going to go into the trial because they're taking IVIG or prednisone. Those aren't ideal. IVIG takes a long time, every month. You got to do this and that. Prednisone has side effects. So if you get a simple subcutaneous injection and an open label extension, you're sort of the winner, and that's going to play into this a little bit too. So all 5 stages had subtle failures which interplayed to all but assure the positive outcome. Much of this, I think, was overlooked by the field and the organizers of the trial. Unfortunately, we may have learned little, if anything at all, about the drug's efficacy. And these are the things that play in: misdiagnosis, criteria selection, the adjudication process, using patient-reported outcomes, incentives to respond or relapse, which is a giant one, reverse enrichment, which I'll tell you about, and ultimately unblinding and inference that goes into stage B, which was the blinded part of the trial. Misdiagnosis in CIDP is a big problem. 2 papers I like to point to are one by Jeff Allen, who's in his expert referral center, getting referrals saying 50% of patients don't have CIDP who are referred to me. The ones who don't have it, they think they responded to therapy. They're talking about objective responses, a lot of subjectivity in responses. The other, which myself and Dr. Barron took place in, was from Todd Levine, and we decided that only 15% of the time could you read a record submitted for a patient who wanted IVIG and convince yourself the patient had CIDP. 85% of the time, that was hard to do. So think of the test the adjudicators are about to have. 85% of the cases are going to be hard to read from our experience. Okay, problem number 2, criteria. Remember, CIDP is not a disease with the gold standard. You cannot just order a test and say a patient has the disease. So you have to rely on criteria. Criteria started with Rick Barron in the late 1980s, but they got usurped by Dave Kornblath, who wrote the top criteria, which are research criteria for diagnosis of chronic inflammatory polyneuropathy. Those are research criteria, so they're specific. They're designed for research. When you look at the patient's attributes, you have to check all the boxes, not just some of them. The criteria, if you read the second paragraph and say, we are willing to miss some patients, but anybody who meets these criteria have CIDP. Those make sense for a research protocol. You're going to get CIDP patients in your trial. What happened in the early 2000s is that people got sort of worried about, these criteria are too strict to use in clinical practice. The insurance companies won't give IVIG to patients because nobody meets the criteria. So the European Academy of Neurology and the Peripheral Nerve Society created a new criteria, which was sensitive. It's designed for clinical practice. You have to check some of the boxes, not all of the boxes. It has to look like CIDP enough, but it avoids not treating patients who really have the disease. And if you read those criteria and look at them closely, it says, you might have to treat somebody and prove they don't respond to therapy to be sure that the patient doesn't have the disease, but we'd rather give everybody a chance first. And that second part of it creates a problem downstream that I told you about at the beginning. Now you're going to be treating people who think they have disease, and they're going to develop a relationship with their meds, even though they don't have it. And before I switch the slide, I just want to say, criteria are not perfect. They're not intended to diagnose CIDP. They're intended to give you an idea about what the patient might have, but there's work to do downstream. And I don't think it's a good idea to pick clinical criteria for a research setting. You're all but asking to have a problem. So the lesson here is sensitive or clinical criteria, by intention, do not eliminate background misdiagnosis as well as research criteria. And in a disease known for misdiagnosis, it is likely that some number of patients who participate won't have the disease. Something got cut off at the bottom, and I'm sorry. Adjudication, the third problem in this trial. And this I won't talk about. You take it for granted, hey, we'll just give these cases to experts, and the experts are going to make a decision, and everything's going to come out great. But if you read a poster that these guys put out after the first 200 patients they adjudicated, and these guys are my good friends who I've talked to about this, so it's not just reading a poster. The protocol says we're going to give it to 2 adjudicators, and they're going to vote, does the patient get into the trial or not? And they looked here at the concordance between those 2 adjudicators, and the concordance between the first 2 adjudicators was 50-50. It was 52-48 to be technical. And some of these cases were easy, so 52-48 doesn't make perfect sense. But it's saying that there was a problem, and they recognized it. And they said, even among experts, it is challenging to accurately identify and diagnose CIDP per the 2010 EFNS-PNS criteria. There was concordance of 52% in the first 200 cases. So to me, this is saying that they had a problem. And having been an adjudicator, having run adjudicator committees, I will tell you, judging these records is not so simple. The records that were made for clinical diagnosis years ago, nobody was thinking about a trial. You don't know if the patients are active or inactive. You're being put into a forced choice situation, so your visceral sensation while you're reading these charts is, I don't really know. I want to say I want to phone a friend, or I want to do this. So it's hard to do. The guidelines that you're using are actually quite vague. I won't go into the details of it, but they're not like you read them and you know what the phenotype is, or the electrodiagnosis have thousands of parameters. It's very hard to do. The records are incomplete. So these guys will tell you this was a very hard process. So the lesson there, it is unlikely the adjudication worked as well as intended. So it is likely that the process enrolled some number of misdiagnosed patients. And by the way, if everybody's guessing, you could get 2 yes guesses in a coin toss just as easily as you could get 2 no guesses, and half as easily as you get a yes and no. So some patients are going to get into this trial who don't have the disease. Okay, so we've done 3 things that are sort of building up the story to what I think is the real problem, but we're going to go to the 4th, which is sort of enormous in my mind, which is the trial uses patient-reported outcome measures. Patient-reported outcome measures have their place, but they're basically, if I wanted to define them for the course of this trial, is a measurement of a patient's health status that is provided directly by the patient without amendment or interpretation by the clinician. So the clinician is not examining the patient and saying, did they get stronger? The clinician is going to a patient who has a relationship possibly with their medicine and saying, how do you feel when I take you off your medicine? And the patient's like, I feel terrible. I can't walk as well. And just that report, they go on to the next stage of the trial. So they're weak. So the message there is in unblinded trial stages, PROs will be exposed, leaving outcomes vulnerable to expectation, incentives, and placebo effects. This differs from blinded trials where the noisy results even out, or from trials that use objective outcome measures where the outcomes are not so easily exposed by this process. So we have a problem. 5th, as you look at this trial design, people wanted to get into that open label extension. I know because I enrolled patients in trial. It's a big incentive for becoming and enrolling in a trial is to get that open label extension. And you have a rule in this trial, which is buried in there, which is when we do the run-in, the stage where we're going to stop their standard of care medicine, they have to relapse. And when we do the next stage where they go on therapy, they have to respond. Only if those 2 things happen do they get to join that open label extension. So take a patient who's one of the more curious, running around, might not have the disease or have active disease. They're going to want to get into the open label extension. They're going to do what they have to do. So the combination of patient-reported outcome labels and an open label extension runs the risk of incentivizing patients to react to the protocol rather than the treatment. Here's a line from a previous trial. So I'm going back to the fingolimod trial, and this is from Dr. Hughes, who's a wonderful British CIDP doctor, and he's listing problems after the trial was negative with CIDP trials. Apparently nobody read this. or somebody read this, and he says, it's difficult to identify participants who still need treatment to prevent deterioration. Although the trial design is aimed to recruit CIDP patients with active disease, about 42% of patients did not have a confirmed relapse when they went on placebo. So people who think they have disease go on placebo, and they don't relapse, so they didn't have the disease. 42% at 3 years, by the way. It's like 33% at 1 year. Didn't have any. So anyway, we go into the run-in, and this run-in, it aims to identify patients with active CIDP. So we're going to stop their standard of care therapy and see what happens to them. So for the rest of this talk, I'm going to show you blue balls and red balls and green balls. The blue ball is active CIDP. The red ball is misdiagnosed patients. And this current trial is looking for patients who might have CIDP but are in remission because their disease is no longer active. So they're going to go on the trial and nothing's going to happen. So we'll call these two categories inactive patients. And the inactive patients, if they enroll in the trial, are going to give you nothing but noise, right? They're just going to do whatever they want to do. And if they have a lot of psychology involved in their reactions, we're going to have a problem. So they stop it, and everybody in the trial relapses when they stop their drug. So the method doesn't work in this type of trial, because if it worked in your perfect world, you'd have this perfect. And why doesn't it work? You have the open label extension that I talked to you about. You have patient-reported outcome measures. Patients are aware they're stopping medicine. So you have people who have this relationship with their drug. I'm just going to stop their medicine. What do you think is going to happen? We know active CIDP patients develop symptoms when their meds are stopped. Anybody who practices sees that. And then there's a certain pressure to the design, because the design is you have 12 weeks to relapse, and you're coming in every couple of weeks, and the doctor's going, hey, you've got 10 weeks left to relapse. I'll see you in 2 weeks. You've got 8 weeks left to relapse. You got 2 weeks left, and if you don't relapse, you're not going to be in the open label extension. Oh, doctor, I can't walk that well. Okay, we just take it. And if you think about it, both sides of that equation, the doctor and the patient, are going to sort of participate to keep that patient in the trial. Again, the comparable number of relapses at 12 weeks in prior trials is about 30% when it's in an unblinded protocol. So this is a very weird result. So what do you think is going on there? I'm going to give you two hypotheses. One I think is unlikely. Only active CIDP patients relapse during the run-in. More than 95% of the participants had active CIDP, and the adjudication committee was given an A++, even though they didn't agree and they were using really loose criteria. I don't think that's the case. I think the more realistic hypothesis is the inactive patients relapse because of incentives, placebo effects, and pressures. The active patients truly began to relapse in this, so everybody relapsed, and no filtering took place at all, so your balls still look just like they did with that 50 to 80% weird CIDP background in the beginning. So no stable patients means no filtering took place. Many inactive patients will enter stage A. Now, stage A, we're going to put everybody back on drug, and we have the same exact problem we had before. Everybody's going to go back on drug, and guess who's going to respond? The patients, guess who's not going to respond? The patients who are absolutely relapsing are not going to respond to the drug. So in stage A, the inactive patients know they are on drug. They're incentivized to respond to reach the open label extension. They tend to stay in the trial. Now you have this reverse enrichment taking place, because the patients who are relapsing, these are my patients. They're going to go into the trial. They're already relapsing because that was the rule. Now they're going to get the research drug. And the relapsing begins, and they start dropping out. And about 30% of patients exit the trial. And CIDP is a hard diagnosis. 30% of patients could potentially be all of the active patients, for all we know. The exit rate is even higher in subjects previously on IVIG. This drug lowers IVIG methods, so they're going to be more apt to respond. Like 40% of the IVIG patients who had previously been on IVIG drop out of the trial at this point. So the active patients relapsed and had to withdraw, possibly to be rescued. Inactive patients were incentivized to remain as it determined eligibility for the open label extension. Entering the double blind, the situation may be worse than it was at baseline, with many patients now with inactive types of disease. So we're used to looking at the next stage, which we're going to get into the double blind. This is how people like to represent double blinds. You go from drug, and then you get randomized to drug or placebo. And in this case, the people who are on drug, they're the responders to stage A, and I hope you're staying with me up to this point. But I'm going to just represent this a little bit differently with 3 pieces. If you look at the left, one group is going from drug to drug. The other group is going to drug to placebo. And the third picture is a curious subject, and doctor for that matter, is taking care of them, who's wondering what just happened to me, because these are sort of shaky individuals in this thing. So they're not, I'm saying the design is in parallel. There's no opportunity to sense a change. And on the other side, there's an opportunity to sense a change. And some percentage of those patients may have had a symptom, and they're going to go to still having a symptom, and some of those patients may have went from a symptom to not having a symptom. The top group is going to further on therapy, and the bottom group might become aware of a disappearance. Like I was joking around with Angela Gengen, my wife, yesterday. Angela told me, take all the crap out of your jacket before you go on a walk. So I took all the crap out of my jacket, and I was like, wow, I feel a lot better. I didn't know. And I say that to you because I went to Angela, thank you, you helped me with my talk, because I wouldn't have reported that I was having an adverse outcome by having too much stuff on my jacket until I knew I took the stuff out of the jacket. And then I knew I could walk better. But I didn't go back to my research coordinator and said, oh, by the way, I was having an adverse outcome. So there's a chance a lot of this stuff is underreported if there's a disappearance. And obviously, if somebody says, I was having an adverse outcome and now I'm not, that might get reported. But there's a sense that there could be underreporting, and this problem might be much worse than they think. One thing that caught my eye in this trial is they had about a 20% injection site reaction that was reported in the open label part of the trial before they randomized them. This number was 40% when the drug was reported for myasthenia gravis, and I'm saying it could theoretically be higher than that. So that leads me to the next part of the discussion, which is there is a lot of psychology about people who've made inferences in trials. We've heard about some of it in placebo group discussions here and other discussions, but there's tons of psychological articles of you put somebody in a trial and let them make an inference. They will not behave the same way if they've been further on drug or not on drug. And there's a lot of terms about it. I'm not a psychologist, so I'm not going to represent to you, but you could get lost in the literature, especially now with ChatGPT. You could get lost in the literature for days, which people will tell you I've done. So with that, the two drugs are in parallel. And if you're making an on-drug inference, your behavior is going to be such that you may think that everything's good and you may not pay attention to your body as much. If you make an on placebo inference, you're thinking, I don't feel well today, I might be relapsing. You've patient-reported outcome measures. So you run the potential here to push the Kaplan-Meier curve up for one side and the Kaplan-Meier curve down for the other side. So that being said, inferences from unblinding may have influenced participant behaviors, in effect particularly problematic given the trial is populated with inactive patients who use PROs. So this is, what I'm wondering about with the trial design is if we've actually invented the Rube Goldberg scheme of trial designs. It feeds on itself. You could study orange juice with this trial design and get a positive effect because you're bringing in a large percent of patients that don't have the active disease and then tipping them off on their therapy. And any tip-off in that patient population could create a problem downstream. So I'm raising the point the result is baked in the cake. And I only have a couple more slides and we'll be finished. But this is back to that Efgar-Tigamont trial, which was a failed trial from about 7 or 8 years ago that informed a lot of the methods that got this trial started. And the treatment group and the placebo group over 3 years behaved exactly the same way. And you could see at the left side of the curve, a lot of patients dropped out right at the beginning of trial, and then there was a subsequent attrition of patients who dropped out. And what's happening at the beginning of the trial is they're stopping their standard of care drug and relapsing. So if we represent the trial this way, and I put up the treatment group of this trial over the same time period, and I just account for things a little differently because I'm accounting. And it's not a perfect accounting because you don't really get to do that when you're reading data from somebody else's trial. But if you look at the patients that dropped out and left the trial over 3 months and then tack onto it what I showed you at the graph at the beginning, because this goes back to when they stopped standard of care. It's not from the start of the later stage. It's from the beginning of the trial when they stopped standard of care. The treatment group looks exactly like the treatment group from that trial. This is the placebo group. So something happened to the placebo group. I think probably that's where the Kaplan-Meier curve got pushed down from getting tipped off. But the placebo group has a dropout rate in this trial, which is somewhat spectacular. So this is the two things drop over that. And this is what the trial would look like if they had sort of mapped these lines from the beginning as best I could tell, where I started 3 months earlier from another view.
Thank you. We probably have time for 2.
So back to criteria. So in the world of John Katz, what criteria should we be using for CIDP studies?
I mean, thank you for an easy question. I mean, you should be using research criteria for research trials. I know what the problem is, right? It's going to be hard to recruit. 2 things that the trials need are, I'll give you 3 things, because that whole first stage, I think, has to be rethought. Research criteria are at least going to give you a much better chance that you don't have all the noise in your trial from a patient who doesn't have the disease standpoint. So it'll eliminate I have a whole nother talk on this sort of risk you're taking by letting patients, because as you get weaker and weaker on the criteria, you're allowing more patients, your specificity is going down. The patients who only meet one thing just might have weird neuropathies that aren't CIDPs, so it's easy. At the top of the criteria, the center of the bullseye, where patients clearly have everything met, demyelination everywhere, typical phenotype, you could have security those patients have CIDPs. So that's number 1. Number 2, the field needs some kind of term for risk of relapse. You can't do these withdrawal stages, they don't work. So you need some kind of predictor that says, if you've relapsed in the last year, it's likely you're going to relapse again. If you've relapsed multiple times, it's likely you're going to relapse again. And from an adjudication standpoint, the field needs to take it much more seriously because adjudication is hard. Just think judges giving people completely different penalties. And from an adjudication standpoint, and from a submission standpoint, we have to work on our, it's very unscientific, but people have to work on their verbiage of how you're going to submit a patient to trial. If I want to tell you, and this is sort of like why Dave Saperstein and I joke about invasion of the body snatchers. If I want to tell you that my patient had a relapse, it's not so easy to do if you think about it. Why would you believe me? I'm just some guy from San Francisco. I lie a lot. So it starts to feel like you're talking to people and you go, I don't trust you. Like, you have to do better with convincing me your patient got weak. Just like when patients tell you they got weak, oh, I don't feel as good, I have to continue my IVIG. It's like, no, I need you to come in, get examined, and see what's going on. Oh, I live too far. All right, I'll just go put you back on IVIG because it's easier. So all these things have to improve, and it would be much harder to enroll. But I do think in an autoimmune disease, if you get active patients, you could probably do very small trials and figure out whether people have disease. Can I just show you guys, do I have time for one more slide, or we're done?
Yeah, we got one more question.
One more question, okay. I'll forget my slide.
Very interesting, thank you. My question was, your analysis requires the unblinding. Do you know if they assessed for unblinding in the study?
Very good question, thank you for asking it. No, they didn't, and I've had specific conversations with them. Did you assess, what was your strategy to assess for unblinding? Did you ask any questions or unblinding? What was your strategy for presenting unblinding? No, pure X's.
One more, okay, one more.
Yeah, I was just looking, can you speak to the objective secondary outcome measures, summed MRC, that would have addressed some of the concerns?
I think that those outcome measures probably help, but I haven't dug down into them as much to answer your question to give it justice. But I do think those measures are also going to be very, as much as they sound objective, they're going to be subject to major subjectivity also. You're just not going to push as hard. Do we trust examiners? It's a very hard question to answer. And grip strength, same problem. Did they squeeze hard enough? Questionnaires, same problem. So I'm not 100% convinced that that would help.
So I think.
Thank you.
Thank you very much. So I'd like to welcome to the stage Suma Babu, who's going to talk about considerations for interpretation of ALS-ASO trial results and the lessons learned from SOD1 and the C9orf72 trials. Thank you.
Good afternoon. I'm speaking at the last session of a rich and loaded 3-day meeting, and I'm going 15 minutes behind schedule. Your minds are full, your coffee cups are probably empty, but you're still here, and I'm really grateful for that. Thank you. Today I'm going to talk about, it's a tale of 2 trials. Both read out negative randomized controlled trial clinical efficacy results, but both had very different endgames. One led to FDA approval of the drug, and the other led to early termination of a program. Let's dissect as to why. These are my disclosures. So the first trial is of the SOD1 ASO in SOD1 ALS ASO, the tofersen trial. SOD1 ALS, as you all probably know, that is a toxic gain of function mutation that leads to ALS, and the fundamental problem there is overproduction of SOD1 protein. Tofersen is, or it used to be called as BIIB067, is an antisense oligonucleotide against the SOD1 mRNA. So the intended purpose of tofersen is to reduce the SOD1 overproduction of the SOD1 protein. When you think about ALS and heterogeneity, we talk about heterogeneity based off of site of onset and various different symptoms. Even within a small population as SOD1 genetic form of ALS, there's a lot of heterogeneity. It accounts for only 2% of all cases of ALS, but there are over 150 or 200 now pathogenic mutations, and even among those 200 pathogenic mutations, as you can see here, the disease duration survival span can be really variable. SOD1A5V has a median survival of 1.2 years, and there are other variants that can have survival span of over 20 years. So when you're running a clinical trial, even for that small genetic subgroup of ALS, with this type of heterogeneity, there are some biases that come into play, and you have to think about trial design and how do we read and interpret those outcomes, and also think about sample size when it is so rare, and how do we enroll those participants in those trials. So this is the trial design of VALOR, which is a pivotal trial of tofersen that included tofersen 100 milligram dosage against placebo. It was designed as a 6-month randomized controlled trial, followed by an open label extension that continued almost up to 4 years for many participants who rolled over from the randomized controlled trial into the open label extension. And these were the primary endpoint was ALS functional rating scale, and in addition to that, there were several secondary endpoints, including SOD1 protein levels, change from baseline, as well as neurofilament change, and several other clinical markers, as you can see here. I want you to really sort of focus on this particular text box. The primary analysis population of this trial was primarily based on fast progressing subgroup that were enrolled in the trial. The definition of fast progressing at the time that the trial was designed was based off of 2 factors. One was the variant itself. There are some variants that are historically known to be extremely aggressive and fast progressing, as I showed in the previous slide. And the second was based off of something called as the ALS functional rating scale pre-slope, that is really based off of recall of participants of how long they've had the disease and how many points they have on the functional rating scale as of today, and how many points they have, how much disability they've accrued over time. So this was the primary analysis data set, and this is important, and I'll tell you why. So when the trial read out, there was target engagement. As you can see here, there was a significant decrease in SOD1 protein within that 6-month period, 6-month randomized control period. This was the first time in the history of ALS clinical trials that a trial showed a really remarkable decrease in neurofilament light chains. Until then, there's not been any other trial that has shown a decrease in neurofilament light chains. And this is, you don't have to squint your eye to see is there an effect or not. It's a huge effect in terms of treatment response and decrease in neurofilament light, suggesting that there is decreased neurodegeneration overall. So this is wonderful so far. And the next slide is where it becomes problematic. 6-month randomized controlled trial, when we look at the ALS functional rating scale, which is a marker of function and clinical endpoint, it did not meet statistical significance at that 6-month time point. And for many of us who have been on the front lines as site investigators, seeing patients, treating patients, it just wasn't making sense because we were seeing clinical stabilization of participants in the trial, but why did it not meet its primary endpoint? And so the p-value was not less than 0.05. It did not achieve, so it was essentially a negative trial. But this is where we started seeing a difference. At that 6-month point, the clinical difference between the groups was starting to emerge, but it was not statistically significant. The open label extension study really saved the day for tofersen. As these participants continued into the open label extension, the real p-value of less than 0.05 started to emerge at week 52. So a huge lag between the NFL change and the clinical change. So the clinical difference, the clinical stabilization difference between active treated and placebo or early treated and delayed treated groups really emerged at that 1-year mark, not at that 6-month mark where the randomized control trial ended, as we didn't know how delayed the effect would be of some treatments like this at the time that the trial was designed. When we looked at additional markers, there was stabilization in almost every other secondary markers. Again, there's a very delayed effect. Even in the delayed start group that started the treatment after the randomized control trial, you see that there is stabilization of clinical markers, whether it's vital capacity or quantitative muscle strength testing. So these are actually really important to think about when you design clinical trials. How long do we treat participants with in a randomized controlled trial design? What are some of the limitations of really fast and expedited, short trial duration? And then what's the importance of open label extension in a disease like this with so much heterogeneity? So the important lessons we learned from this trial, there are several important lessons the tofersen trial has taught us, taught our field. The most important one being that the clinical stabilization really lags behind biological change. And so tofersen did decrease NFL, and it was really important that we saw that huge decrease, and we didn't kill the project at that time because it was a negative study, but we continued on and looked at the long-term outcomes of these individuals, and we saw a really remarkable stabilization of clinical function as well as survival in these individuals. So clinical stabilization lagging behind biological change is an important consideration while designing clinical trials. And then the other important lesson is that when you see a p greater than 0.05 in a randomized controlled trial, we all worship the p of less than 0.05, but sometimes it's not really statistical insignificance because the drug doesn't work. Sometimes it could be because of the trial design itself. And it's not because the trial design was a poor design. At the time, we didn't know how long it takes to actually see a clinical effect. So p of greater than 0.05, it's important to put things in perspective with the overall picture before killing a project. And it's important to also think about how we can de-risk these trial designs in early phase trials. It's really important to think about not just for ASO products, but other experimental products in ALS, about what is the right treatment duration, and in a later phase clinical efficacy testing, can we de-risk this in an early phase trial to understand how soon we can see a biological change, and can we incorporate open label extension into those trial designs in early phase trials as well to look at early signal of clinical efficacy before embarking on large phase 2 or phase 3 trials. And then it's important to think about also what the end game is. And oftentimes, when we design clinical trials, it's limited by budget. We want those, really fast answers. But if the end game is to understand whether this is a drug that potentially can cause clinical benefit, do people live longer, do better on this drug? It's important to incorporate open label extension, and even in some programs to think about expanded access as well, because these additional programs can offer a wealth of information that the randomized controlled trial itself may not offer for a really heterogeneous and difficult disease such as ALS. So in retrospect, did we do the right decision by bringing this drug forward and getting the FDA approval and treating participants? This is new data that has emerged from the open label extension past it's almost 4-year open label extension data. And what you can see here is there's really beautiful stabilization of ALSFRS-R, vital capacity, as well as quantitative muscle strength testing in those participants who continued on tofersen in the long-term open label extension beyond even that 52-week mark that led to the FDA approval. I want you to also pay attention to the text box that's highlighted in red at the bottom. The numbers start to dwindle down as you start from the beginning all the way to year 4. And that's important to think about as well. I mentioned earlier that even within SOD1, there are really aggressive forms of ALS, and then there are very slow progressing forms of ALS as well. When you see numbers starting to dwindle down like this, you want to think about the fast progressing forms that potentially these participants are progressing and their disease is running on a really rapid train, and it's really difficult to catch those individuals and stabilize the disease. And so the stabilization that you're seeing here is probably mostly driven by slow progressors, and that's important. That's another lesson. We'll talk about that in just a little bit. It's the push and pull between, do you incorporate slow progressors in clinical trials, or do you really focus just on fast progressors in clinical trials? So there is that push and pull between statistical modeling. When you have fast progressors in a trial, you can design a trial with adequate power with shorter treatment durations with the assumption that you're going to see a clinical signal over short treatment durations. But the important aspect that we learned from this trial is that it's also important to have slow progressors in trials, because it's really slow progressors that showed maximum stabilization of function and survival in the long-term open label extension of tofersen trial. When you have really fast progressing individuals in a clinical trial cohort, their disability accrues very fast, and there are mortality risks in this population. Even if you see a biological change, even if you see a 70% reduction in NFL on an individual level, by the time you see the clinical change, which really lags behind by almost 12 to 16 weeks after the NFL change, by that time they've really accrued a lot of disability to see a meaningful clinical impact at that point in time. So the statistical power boost, does it really outweigh the risk of this extreme fast progression in clinical decline? I think is another question we want to think about. And then when you're enriching trial cohorts with really fast progressing individuals, it's really important to have that early intervention, maybe very early, maybe super early, maybe at the pre-symptomatic, at-risk stage, even before the first symptom of weakness has emerged. And that's important to consider, especially if you're focusing on fast progressing individuals, because the window of intervention can be really short in some of these individuals. Moving on to the C9 ASO, this is a completely different program, and BIB78 targeted one of 3 or 4 different pathological mechanisms in C9 ALS, which targeted the sense strand RNA. And as you've seen in this complex slide here, C9 genetic biology and disease mechanisms are complex. It's not just a toxic gain of function, but there's also haploinsufficiency that contribute to disease. Which one takes predominance over another, we don't know that yet. But it's important to think about all mechanisms while designing a trial and not just one aspect of it. This was a trial design. This was a phase 1 study. The numbers overall were very similar to the tofersen trial. Tofersen trial had 108 participants, and this had 106 participants. The treatment duration was very similar, 6-month-long study. Each cohort had even smaller numbers because this is a genetic condition, and it can take a while to accrue participants and enroll participants in this disease. So we're focusing on the last cohort, which had the highest dosage versus placebo. You're looking at numbers in the teens. And so this is really a comparison of those numbers. primary endpoint here was mostly safety tolerability, unlike tofersen, it was looking at functional rating scale in the VALOR study. And these were the results. The target engagement, really beautiful target engagement in this phase 1 study. Even in those small numbers in the teens, we were able to achieve decrease in dipeptide repeats. Poly-GP is a dipeptide repeat. In the treated groups, the solid, basically, that's at the bottom, is looking at 2 different dosages, 60 and 90 milligrams. Decrease compared to the dotted lines, placebo at the top. And same for poly-GA is another dipeptide repeat. Again, beautiful target engagement in the CNS. When we looked at the neurofilament, the NFL, this was a completely different story. There was an increase in NFL in this trial, a 36% increase in NFL in the BIB78 treated group. When you start seeing trends of active treated group showing worsening compared to placebo, it is important to also look at the clinical markers. When you have numbers in the teens in terms of sample size, just an increase in NFL alone may not be sufficient to interpret this completely. When we looked at ALSFRS-R as well as vital capacity, everything was trending in the unfavorable direction for the treated group. And I really applaud Biogen for making that difficult decision of early terminating this program at the right time before exposing more individuals to a drug that potentially could cause harm. And this was a very different outcome compared to the tofersen trial outcome, despite having a negative read on the randomized control trial. But I want to also think about C9 as a disease biology, and I think as a field, we all know how to interpret a huge decrease in NFL. A huge decrease in NFL, we all have unanimous approach of interpreting this as, yes, the drug has benefit, because otherwise NFL would remain stable throughout the disease course. But as a field, do we really know how to interpret increases in NFL? Does it always mean that the drug is causing harm, or could there be alternate explanations to this? I think it's really important for us to think about this as we design more clinical trials, and it's also important to think about the genetic subgroups we're targeting for those trials when we're interpreting NFL changes. For example, in C9 ALS, we know that it is not just a disease of motor cortex or motor neurons. There is also an involvement of frontal and temporal lobes that can arise at any time during the disease course. And this PET imaging, TSP or PET imaging from our group shows that there is an increased PET signal in the motor cortex as well as in the frontal and temporal lobes. Have we paused and wondered about what are those lobes actually contributing in terms of NFL to the big picture? Do we know about this? NFL is non-localizing. We don't know that. Do we know the kinetics of NFL? Where is it coming from? Is it all coming from the motor neurons? Is it coming from other parts of the CNS? Is it coming from peripheral nervous system? We don't know that. So I think it's really important, especially when we design smaller clinical trials with small sample sizes. There is variability, and there are other considerations to think about when we see an increase. So the clinical context is really important. And then when you think about the NFL increase in other programs, we look at Zolgensma. Zolgensma and SMA showed a huge increase in NFL. The levels went up from the 30s, 40s to almost 200 and almost close to 300 within a 1-month period. And then it came back down afterwards. But we know that Zolgensma is clinically effective. So a rise in NFL is really harmful. Maybe there's an alternate explanation to that rise in NFL. And then what we know about C9 ALS is that there is an age-related increase in NFL as well. When we're enrolling participants in a C9 trial that spans age groups of 30s, 40s, 50s, 60s, and 70s, this is important to keep in mind as well in terms of the overall data set and how that might impact the NFL changes if we're looking at NFL as an outcome. What we've learned is that there's about a 4% age-related increase in NFL greater than 50 years of age. So do we really know how to interpret an increase in NFL in small sample size clinical trials? I think this is a very important question that we have to think about as a field. So with that, I really want to acknowledge the tofersen development group and Steph Fredette for providing me with some of the slides from both of these trials. Steph Fredette is at Biogen, and I'm thankful to our group at the Mass General and the Healy Center for allowing us to be a part of both of these programs and offering these treatments to our patients. Thank you so much.
So, thank you. That was a fantastic talk. I think, given the delay, whilst our next speaker comes up, we could take one question. I think Valeria's at the front, and perhaps I think that was the only, yeah.
Thank you, very nice talk. I just wanted to comment on the SMA, because that's a transitory increase, and the kids are doing very well, and it's transitory. When you add an add-on drug, there's a study ongoing, the neurofilaments go back down, and so the interpretation is that it may be an inflammatory response. And maybe the question is not just that the neurofilaments rise, how do we interpret it, but what is the sense of the neurofilaments? In SMA adults, for instance, neurofilaments are low to begin with. So, and there's neurodegeneration. And that won't be useful to monitor, right? So.
Thank you for that comment. I think that ties to the same theme of, in order to interpret NFL, we really have to understand the context a little bit better, both in terms of the drug, the trial, the disease population, the demographics of the disease population.
Fantastic. I encourage if anyone has any further questions, and perhaps they can find you at lunch to discuss it further. Thank you.
Thank you so much.
So I'd like to invite to the stage Professor Lauren Surveys, who will be talking about Translana and beyond, and from the University of Oxford, among others.
Thank you very much. I realized that I'm the last thing between you and a nice walk in a sunny, walk around the lake. That's actually the title that I was given, Outcome Measures and How to Approach Regulatory Requirements, Lesson Learned. And that's me preparing that talk, and there is a scale that has been developed for speakers that we call the motivation meter. And it goes from, this is going to be the best talk in the history, to, I will say that I have COVID and I can come. I was actually scoring to, I don't like the title. So I proposed a new title, which is Assessing Swallowing Difficulties with Belgian Beers, a New Outcome for Bulbar Dysfunction. And unfortunately, Emma did not buy it. So then I came here 2 days ago, and I discovered in the program that the title was absolutely not the title that I was preparing my talk for years. And Translana and beyond, is it a joke? So I say, Translana, are you joking or what? You want me to conclude this meeting on Translana? Is it a joke or what? Come on. So Valeria told me, no, it's about caution in interpretation. You can even be provocative. Have you really said I can be provocative? You say that? You really say that? Well, as long as you behave. Okay. I've got a couple of disclosure. And I will start with a kind of email that maybe many of us have received, but to be honest, I receive it like every day. Dear Professor, my name is and we just learned that our son Dylan, who is 5 years old, has DMD. We are devastated and we wish to do whatever is possible. We have learned that you have a trial with a treatment that stops the disease. Please, doctor, please accept our son in the research. You are ready to relocate close to your hospital and do whatever is needed. It's the only chance for Dylan. Thank you, doctor. Thank you. My wife and I are praying for you every day. We receive this type of email all the time, and I see that, some facial expression in the room. I'm obviously not the only one to receive this kind of heartbreaking emails that make you understand that people have not understood what we're doing. And this leads to several phenomena. Relocation. I mean, we all have patients who relocated, who changed their life, and who took actually some very difficult decisions for the family, for themselves, for the patient, but also for the siblings. Crowdfunding a lottery is something that we have observed over years. Crowdfunding is a big issue. I don't know if it happens also in the US, but we have a lot of Polish patients, a lot of patients from India, crowdfunding and trying to get access to trials, not to treatment, but actually to trials. And it comes with a lot of questions from an ethical perspective. But for me, the main question is that we actually put the kits on the market and we decide which kit will be funded and which kit will not be funded. But on which basis? Is it on the basis of the treatment that could help the kid the most? No, most of the time it's because the crowdfunding campaign is well conducted with a mom with nice looking, with happy to cry and to laugh at the right moment, with middle class, with like us, with white, and we know that there are plenty of factors that will influence the success of crowdfunding. And this brings a problem of equality. Is it the way we understand, we as Europeans, the social security? Just to put the kids on the market, to vote for and cons, and to have a kind of Hunger Games? Personally, I don't think so. If you don't like lottery, there is another nice mechanism that we call, if you don't like crowdfunding, there is another mechanism that we call lottery, in which we actually randomize patients to get access to an approved treatment for free. And that's what happened, and that has led to a lot of issues, I can tell you. All of these problems are coming, in my opinion, from the same mechanism. It's the fact that we actually raise expectations and raise hope in the community that are not reasonable, and that we bring in the head of the patients, in the head of the community, a potential benefit that we're not going to achieve. So the objective of this talk is that to convince 50% of the audience that all of us, not just the bad pharma company, the stupid doctors, but all of us, we have a part of responsibility in this situation. And my second objective is to convince 25% of the audience to adopt one of the proposals at the end of this talk. That's what I want to do today. I will not throw anyone under the bus. I swear it. I will have to take some examples, from what we've lived, but it's nothing negative against anyone. And the only person I will try to throw under the bus is myself, because I consider that I have a part of responsibility in this overstatement and in raising unreasonable expectations. I have a part of responsibility. I take it. I will take, several examples, and I can even state what I've done wrong in these stories. So first problem is the interference of other medications and patients' training. Can I have the video, guys? This is a patient with Duchenne on the left side before the treatment, on the right side after the treatment. And as you can see, after the treatment, he started after, but he's going to go much faster. He does not use the banister. He's able to alternate the steps, and now he is, with several seconds in advance. So we have a treatment that works, right? Is it gene therapy, cell therapy? No, it's steroids. It's steroids. This kid has been given steroids. It means that when we give steroids to our Duchenne patients, 3 months or 6 months a trial, if we modify the scheme of the steroids and go from a weekend treatment to an everyday treatment, we are generating an effect that has nothing to do with the treatment that we actually study. You probably know this trial of microdystrophin and the huge effect that has been observed in 4 patients, right? I'm talking about 9 points on the North Star. 9 points on the North Star is big, it's huge. It's something that even a blind medical student is able to see. So, yeah, it's true. But the reality is that when you come to a phase 3 trial with a double-blind randomized placebo control, you do not observe this anymore. Why? Well, this is also something we have observed in other gene therapy trial. This was from another gene therapy product, the phase 1 trial. Look, the difference on a propensity match analysis was 7.5 points on the North Star, never reproduced in a double-blind randomized placebo-controlled study. The problem is, if you are the father, if you are the mother of a patient with Duchenne, and you see such a phase 1 trial, you will do anything to get access to the treatment. You will relocate, you will crowdfund, you will try to make any decision that will help your child to go to the trial. And if we're not able to reproduce it, if we have just overstated our data, we are generating more problems than solution for our patients and for the community. Propensity score analysis for me is a very interesting feature in our field. Have you ever seen one that is negative? I only see positive propensity match analysis score. Why? First, because there is a bias of selection. We just see those that are positive. But B, also, have you ever seen a pre-established statistical analysis plan hierarchizing the outcome, or all of a sudden we see something positive, but we don't know how many outcomes have been tested before we get one that is positive. So we need to be very cautious when we interpret this data. And as you can see, this data of the first trial that I've shown with another product, the patients were there. So it's not a surprise that they were increasing. They were at the part of the natural history study where they are increasing, especially if you give them a lot of steroids, especially if you train them to make the test. So we have the same problem in SMA. These are patients treated with nusinersen, and as you can see, in the early phase, they increase, they improve, and then they start declining. When we start an add-on therapies in these patients, where are we? What are the patients that we're considering? This is from an add-on therapy with SMA. Those patients are treated with nusinersen. We give them an add-on therapy. The problem is not which one. We give an add-on therapy to these patients, and look, they improve by 4-5 points on the HFMSE. That's magic, right? But the problem is that when we try to reproduce it in a double-blind randomized placebo-controlled study, we have a difference, and it's great, and it means a lot, but the difference and the improvement of the patients on treatment is 0.6. And if I want to put the two lines together, this is the phase 2, this is the phase 3, right? Nothing in common. Why? Because in the phase 2, we had a lot of patients from here, and in the phase 3, we had a lot of patients from there. Right? And I'm not saying this is not interesting results, but I'm saying that if we show this to families all over the world, they will relocate, they will make the wrong decision for their family, and they will overestimate the potential impact of the therapy. So my message is, I've shown these results, I've shown this video several times, and I feel that I'm part of the problem. But we should all feel that we are part of this problem and really think about when we are considering efficacy. Well, you may remember this paper in the New England Journal of Medicine with the gene therapy for spinal muscular atrophy. And these two patients, and I've shown this video myself in some of my talk. Here is one of these two MAGIC patients. It's not that one, but it's that one. It is one of the two super responder from the New England Journal of Medicine that was supposed to present patients with SMA type 1. If I'm the father of a patient with SMA type 1, if I'm the mother of a patient with SMA type 1, and I see this video, I will relocate, I will do anything to get access for my kid to this type of treatment. And I will say, oh, we have other drugs that do not work. I want the drug that cure. But what we know today is that these patients were identified by screening. They were not identified per symptoms. So it just means that it's fantastic. We can be game-changing if we treat early. That's a very positive message, but that's not a SMA type 1 patients. And if we claim that it's a SMA type 1 patients, then we generate all these issues. We generate the relocation, we generate the crowdfunding, and we generate the lottery, and we generate irrational aspect from the family. There is another problem that we are facing every time that we try to interpret a posteriori this type of results. It's something that we call the paradox of Simpson. Do you know what it is? No. The syndrome of Servet type 1 is a rare condition that affects midlife individuals and that is characterized by hyperactivity and chocolate hyperconsumption. It's Servet type 1. To try to treat this terrible condition, researchers tried a new injectable protein, the chafalonine. The chafalonine has given encouraging results in phase 2 studies, and researchers now start a phase 3 that includes 1,000 patients in the treated and 1,000 patients in the untreated group. The good news is that we have the perfect outcome. It is the revised, expanded, revisited Mercury scale, the area MS. It gives a point, how many countries do you visit per week, how many miles do you walk per day, how many hours per day are you awake, and you get a score, right? And normal mentally healthy individuals score between 10 and 20, and affected individuals score above 30 in several cases. Let's run a double-blind randomized placebo-controlled study. Groups are well balanced. The score was 40 plus or minus 5 points in treated patients and 41 plus or minus 6 points in the placebo. Perfect. We have the double-blind randomized placebo-controlled study of the century. And after 1 year, wow, the placebo are doing worst. Well, because improvement goes that direction on the revised, expanded, revised, and revisited scale of Mercury, and the treated goes that way, right? It is highly significant. We can finally treat Servet type 1 syndrome with a cefalonin injection. Champagne. But let's now stratify our samples. So if we stratify our patients by BMI, no problem. If we stratify by the severity on the revisited expanded scale of Mercury, no problem. If we consider the age, no problem. Oh, what happens? In female, the drug does not work at all. It's even worse. So in male, it must work very well. So we have a drug that works. We have a drug that works in all patients, but it doesn't work in male and it doesn't work in female. What's happening here? It's what we call the Simpson paradox. Actually, look, if we have a story in which the patients, the male patients, for instance, tend naturally to be worse, and female who tends naturally to go better, and if your groups are not balanced, for this parameter that you decided to ignore in your stratification, you can build an effect that is positive for the entire group, but that is negative for every single of the subgroup. That's what we call the Simpson paradox. So it means that if we start treating a disease that we do not know, if we do not stratify properly our population, we can expose ourselves to the Simpson paradox. Be careful to this guy. And do we have examples? I do have examples. And this brings me back to Ataluren. This is the registry comparing patients with, for loss of ambulation in patients who have been treated with Ataluren and, a control group. But the control group has absolutely not been balanced by the gene mutations, and we are actually comparing with the entire population, and we are matching for all criteria that has nothing to do with the problem. And interestingly, we observe an effect that is longer than the duration in the trial because there were other matching issues. So by not matching properly our patients, by not stratifying our patients properly, we can artificially construct Simpson paradox. Okay, we, the pharma, the authors, the speakers, the reviewers, the attendees, the readers, we are all responsible, including myself. But also the patient's advocacy group. You know the magic story of X-linked myotubular myopathy. We have something that works. This is the most severe condition I've ever seen in my bloody life. But we have a gene therapy that works, and these patients are doing better. It was published. But the problem is that like 23% of these patients died because of the gene therapy. Then we have an ethical issue. Maybe it's worse, but we should maybe think about that. But the fact is that the trial is on hold. On the very same time, and we are still in 2025, what happened to Jules was transformed thanks to the Téléthon. Jules was treated for the myotubular myopathy, so now he's doing perfectly fine. We are in 2025, and there is no mention about the fact that the trial has been stopped. There is no mention on the fact that there were kids who died out of this. What will happen the day after the show? I will receive hundreds of emails of patients who tell me, doctor, I want the drug that works, I want the drug that cures. And these patients will relocate, and these patients will crowdfund. So we are all responsible, we as investigators, you as a pharma, and also the patient's advocacy group. So I have 8 proposals that are all easy and a dream, which is just a dream. So first, let's agree that a phase 1 is a phase 1. And the aim of a phase 1 is safety. And we should present our data of safety, like safety and nothing else. Let's consider post-hoc analysis with external control group only when pre-established statistical analysis plan is made available before the analysis is done. After that, it's too late. Let's not give sponsor a rest until they publish negative results. When we are the presenter of a phase 1 study, let's present it as a phase 1, just a phase 1. When we listen to a presentation of overstated phase 1, let's just react politely and say, fantastic talk, thank you, but please, this is a phase 1. Take it as it is. When we write, when we publish, when we review, when we read a phase 1 study, let's be clear, it's a phase 1 study, let's react. We can be polite, friendly, politically correct, and say, this is a phase 1 study, please keep it as it is. Video are super helpful. I'm addicted to video and to show videos of my patients, but let's be sincere. They are always the best and the most exemplative care. And we need to acknowledge that. And we need to be extremely careful with the expectation that we raise with video. And then let's work with patients advocacy group for helping them not to amplify our overstatement. This is extremely important because advocacy group and patients group is the primary source of information of patients. And my dream, and this is much more difficult to achieve, would be that data should not be made public until peer-reviewed. And I appreciate that when a company is rated on the stock exchange, the information has to go out and it has to be available to everyone at the same moment. That's the ethic of the stock exchange. But the problem is that we are substituting the ethic of research, the ethic of medicine, and the ethic for our patients to the ethic of the stock market. And I think that we should really have a deep thought about that. We need to think about that for the father and the mother of Dylan. We need to think about that for the father and the mother of all the patients written every day. And we need to acknowledge that by overstating our data, we are generating big issues for many families. And with that, I want to thank my fantastic team in Oxford and my fantastic team in Belgium, and I hope I've been provocative enough. Thank you so much.
Thank you. That was a fantastic talk. I think unless there are any questions from the audience, then perhaps I can encourage you to discuss it and make your plans to make that dream a reality over lunch.
Can I just ask one? So that was awesome. That was such a fantastic talk. And I don't study muscular dystrophy, but I am in myasthenia, and I know about the pre-release of data and some of the points that you put up there. And I apologize if I'm naive, but have you published your methods and your wishes? Because I think this would be so awesome to share with our community, but also with students and residents and folks that are going to be doing this research.
Thank you.
So just before you go, I think I'm right in saying that James will announce the Shark Tank winner.
I don't know if the presenters want to just come up, because you've each got a small gift to be given, and then we'll announce the winner. So do you mind coming to the front, the four presenters? And whilst you're... Are they here? They're not left already. Oh, yeah. So, I mean, I guess the first thing to say is that so the four people that presented, there were 30 abstracts. So, you know, even getting to this point, I think everyone is already a winner in many ways. And I was certainly very impressed by the presentations. There were collaborations happening sort of just as we were talking. And I think that's really the most important thing. And hopefully you found that sort of process really helpful for your projects. Sorry, I know you feel like you just stood in front of the whole audience now. And as I say, one person gets the money, but everyone gets an offer of mentorship from the NMSG, and hopefully everyone's found that a really useful experience. So without further ado, the Sharks had a lot of back and forth. The audience votes were taken into account. And I'd like to congratulate Grace from Belfast for winning the Shark Tank.
So maybe a round of applause for all of the... So I think we're at the end of the meeting. I'll just say thank you all for all your fantastic contributions and participation throughout the whole 2.5 days, although it feels like 3 weeks. But it's been great, and we look forward to seeing you all next year in San Antonio. So, Rick?
No, it's good. Thank you. I don't really have much to add. It's been an incredible meeting. Thanks again to Emma and Valeria. Of course, thanks to Liz and to Donna for getting this all together, and thanks to all of you for coming and showing up. It still amazes me how our enrollment keeps getting bigger year after year, and I keep wondering, why do they keep coming back? But I'm sure there's a lot of good reasons. So thanks for coming back. We'll see you next year.